Good morning, everyone, and thank you for being here with us on this Saturday morning for the update on the Center for Infectious Disease research activities. I would like to thank you for taking some of your time to be with us. I would like to welcome the Dean of the Faculty of Medicine, Dr. Raymond Sawaya, and also I would like to welcome Dr. Fadi Daniel, who is representing the President of the Physician Syndicate in Lebanon. And welcome to all the faculty, residents, fellows, students, and research assistants, some of whom will be sharing with us their research work. The Center for Infectious Diseases Research is basically a voluntary effort by interested faculty members, those of us who are interested in research on infectious diseases, microbiology, and other related fields that are impacted by microbes or by infection. And the faculty members who are in the CIDR are from several departments and from a couple of faculties, medicine and the health sciences, and from the departments of pediatrics and medicine, the divisions of infectious diseases, from the pathology and lab medicine, clinical microbiology, as well as basic microbiology, immunology, and pathology. So the CIDR started as a smaller facility. It was called the Infectious Disease Research Core Facility, which was initiated with funding from the Department of Defense in the USA. So we bought some core equipment to do more advanced infectious disease research, and then a few years after that, we got funding from the Dean's Office at the AUB to establish CIDR as a collaboration. It was just seed funding, so that then we were on our own. So currently the CIDR is a standalone entity that is funded by grants that we get as faculty members, and partially by some service tests that we do that generate a little bit of income to the center. So in the next few hours, we have a busy agenda that will tell you about different activities that our faculty members, our research assistants are doing in areas that span the spectrum from basic immunology to cancer. And of course, a lot of infectious disease and microbiology in between. So the first half will be covered by mostly faculty members from CIDR, and then the second half will be presentations by research fellows and research assistants who are working at the CIDR. We have been busy trying to get grants to support our research activities. We've been fortunate to have funding from the Fondation de France to fund our global influenza hospital surveillance network. Since 2018, we get annual funding to do respiratory virus surveillance at several hospitals in Lebanon. We also have funding for investigating invasive meningococcal disease in collaboration with the Ministry of Health. Funding also to develop an assay to identify invasive fungal infections by PCR, and we'll have a presentation about that. We also had some funding from the for one of our students who will be presenting about nasopharyngeal carnage of Streptococcus pneumonia in children younger than 5 years of age at various schools and daycare centers and clinics in Lebanon.
We also have funding for a similar study on Staph aureus carriage. We also have major grants for Dr. Matar and Antoine Abu Fayyad to support research on molecular identification of antimicrobial resistance and monitoring, and also evaluation of nucleic acid test kit for confirmation of cholera infection, where Dr. Abu Fayyad and Matar have become regional and global references for the research on cholera. Also, Dr. Yasser Hal has a couple of research funds studying the role of EBV in inflammatory bowel disease and inflammatory reactions. We also have a collaboration with the WHO for genomic surveillance of SARS-CoV-2, and also another fund to investigate the microbiome in acute viral diarrhea. So these grants are what keeps us going, but we also provide training activities that are delivered to various health sectors. Dr. Antoine Abu Fayyad has been a leader on these training activity. He has been contracted by the WHO to deliver training for cholera specifically in various countries, very challenging to visit countries like Afghanistan and Yemen and South Sudan and Sudan. You know, cholera shows up where there is turmoil, political, social, and upheavals, population movements, etc. So he's been all over the hotspots of the globe to deliver these training activities. And also locally, he's done a lot of training for sequencing and genomic analysis of influenza, and was supported also by Dr. Fatima Dakrub, our lead postdoc at CIDR, who has delivered training for the National Influenza Center and the Epidemiology Surveillance Unit at the Ministry of Health, and other training activities where we were on the receiving side. So we had our staff go to get training in different areas, different parts of the world. Dr. Fatima Dakrub went all the way to Vietnam to learn about viral genomics and bioinformatics. She also went to the UK at the Sanger Institute to receive some training there in genomics as well, and several other activities that were both local and international that were received by our trainees. So capacity building at our center for our staff to just improve our ability to do top-notch research goes hand in hand with trying to obtain funding to keep things going. One of our recent achievements was the development of two vaccinology online courses, the advanced course and a fundamental course. This was developed in collaboration with AUB Online. These are purely online courses that address everything about vaccines, how they are developed, how they are regulated, how they are registered, about all the vaccine preventable diseases, whether they have currently vaccines or they have vaccines in development, and as well all the challenging topics such as vaccine hesitancy that is now becoming a major challenge globally. And we have CME credits for these courses, 72 for the advanced and 42 for the fundamental course. And we have 46 global speakers. About 1/3 of these speakers are CIDR members, but the rest come from the WHO, from Gates Foundation, from the USA, from Europe, from India, from South Africa, from basically all over the world. Every speaker is specialized in their particular area where they present. So it's really a top-notch program. And at the end of these courses, the learners will get a certificate either in advanced vaccinology or fundamental vaccinology. And hopefully we'll get all our fellows trained in this. I know all the pediatric fellows have registered for these courses, and I think the adult ID are on the way as well. So this will have a major public health impact, because globally the vaccinology courses that are available are mostly in person. So anybody who wants to be trained in vaccinology has to dedicate, some time to go somewhere to attend a 1 or 2 week course. So this is the only fully online course globally. So we have a unique niche that hopefully will attract learners from all over the world. And indeed, we just launched these courses this month, about 2 weeks ago. And we have the first batch of learners. We have people from Somalia, from Ethiopia, from Iraq, from Jordan, and of course from Lebanon. I won't spend too much time on our research activities because you'll go into details in some of those activities. One of our busier projects is the Global Influenza Hospital Surveillance Network, where we have 6 other hospitals in addition to AUB that participate in this surveillance in Lebanon. There are 27 or 28 global centers as well, and all are coordinated in France, where all the data is collected. It's available globally, and data for the sequences of influenza viruses that we detect are shared with the WHO to help decide on what vaccine to be prepared for the coming season. So this is something that we are very proud of achieving. And the hospitals that collaborate with us in Lebanon are representative of the different governorates of Lebanon. So we have in the north, we have Mazloum Hospital, we have the Beqa Hospital, we have Hamoud Hospital in the south, and Kisserwan Medical Center in Mount Lebanon, and 2 or 3 hospitals in Beirut and the larger Beirut area. So this is some of the data that we get. So these are the respiratory viruses that we look at, mostly influenza, RSV, and SARS-CoV, and we do screening throughout the year. We enroll patients who are hospitalized, and we hospitalize for specific diagnoses, and we screen them for these viruses, and we collect all the data about their morbidities, what happens to them, their outcomes, etc. This is what we saw this past season, or this current season. We've seen a lot of influenza A, mostly H3N2 activity, that started quite early in late October and is still going on. We do sequencing for a lot of these isolates, so we can characterize the viral isolates at the molecular level. This is the data about the few H1N1 strains that we got. Most of the isolates actually were H3N2, the subclade K, which is wreaking havoc across the globe because of 9 new mutations that occurred in critical domains of the virus that have caused immune escape and a more virulent infection. So this is something that we are contributing to quite well. An exciting project that we have in collaboration with the Faculty of Arts and Sciences is to develop this viral map of Lebanon, where we are plotting this data in real time, and we'd like to have it available for everybody to see and to share very quickly. So these are some screenshots from what the virus would, what the viral map of Lebanon would look like. And I'll show you a draft of the link. This is some of the data that we get. Number of hospitalizations, age, time, peaks, etc. This is the draft link here. We'll try to open it. Just to give you a brief flavor of what to expect from this map. All right, so that's how it starts, but you can, this is back in 2019. So we plotted data in the past. We can plot the data by week or by month and show at what month, what were the activities. This is, for example, November 2020. It was all COVID. We had very little influenza. If you remember, that was in the midst of the COVID pandemic. Then more recently, let's say October 2022, it was all RSV. More recent, we had in August 2024, the summer was mostly SARS-CoV-2. In April of 2025, we had a lot of RSV, which was a bit atypical. And more recently, we have RSV and SARS-CoV. So this is some of the data. We can also look by seasons. So every season, you can see what viruses showed up. So that data is available in more detail. And you can also click at the different centers that are cooperating and get specific data for that region. And there are a lot of statistics that you can get, and we're still working on fine-tuning these so that it will be a useful resource for people who are interested in this. Can we go back? Thank you. So this, I think, is my last slide, just to tell you about projects that are ongoing that we will not be discussing today. So we have this study in children about the Staph aureus carriage, which is an important point to learn about how prevalent is Staph aureus and particularly MRSA carriage in our population. We're looking at the comparative sensitivity of definitions that try to identify people with influenza-like illness or with severe acute respiratory infections, because we observed that there are many challenges in identifying all patients who come in with these respiratory infections. We're trying to see whether 16S identification of pathogens in patients with febrile neutropenia would be helpful in detecting organisms before cultures. We're also working with the AUST to screen patients with cancer by using their breath to look at volatile organic compounds. And we just had a presentation a couple of days ago. The data is very exciting. This is a bit far away from infection, but you have to remember that infection, immunity, and cancer are very interrelated. And the next step will be for us to look at the role of these volatile organic compounds in various infections. We're also looking at Klebsiella pneumoniae, trying to characterize it genomically and try to identify the serotypes of Klebsiella that are causing invasive disease, because Klebsiella is one of the prioritized pathogens for vaccine development by the WHO. We're also reviewing our experience in MRSA, methicillin-resistant Staph aureus infection in hospitalized patients, because we have no clear idea about the burden of these infections. Also reviewing our infectious complications experience in patients who received, pediatric patients who received hematopoietic stem cell transplantation. We're looking at the diagnostic value of molecular testing on pleural fluid in patients with complicated pneumonia. We're trying to learn more about our experience with mycoplasma pneumonia infections. We have the survey about SCID newborn screening. You know, we run a national program for SCID screening for newborns, but we feel that there is a lack of awareness among many parents and healthcare workers, so we're trying to assess that. And we're looking at the nasopharyngeal carriage of strep pneumonia and sickle cell patients, and also looking at our group B strep experience, maternal carriage and neonatal infection, because this is another candidate for vaccine development, and looking at our experience in GI panels. So I'll stop here. This is the last picture we got from January 2024 when we had our last meeting, and next time we'll put the picture from today. So thank you very much. Dr. Ghassan Matar will now share with you our experience as a WHO collaborating center.
Hello, everyone. I don't want to take too much time, but I would like to add about the designation of the CIDR and the Department of Experimental Pathology, Immunology, and Microbiology as a WHO collaborating center for reference and research on bacterial pathogens. So this shows the designation that occurred in 2019, and mainly the terms of reference are to support WHO in monitoring, tracking, and combating, and especially on antimicrobial resistance through research and training. It serves also as a training center on phenotypic and molecular diagnostics of bacterial pathogens, and also it provides a laboratory diagnostic support at regional or global levels. So the mission is to serve as a national and regional hub for microbial genomics, AMR, and outbreak response. And since the designation, we provided the critical technical and scientific leadership to support public health decision-making across the Eastern Mediterranean region and the African region. Now, the core functions are molecular typing, whole genome sequencing of priority bacterial pathogens, regional capacity building through WHO, GORM, GTFCC, MSF, CDC, and MFNet training programs that we have provided in our center, and integration of genomic epidemiology with public health action, along with AMR surveillance and outbreak response coordination. So in that center, actually, we have PulseNet that was formed before the WHO, CC, and CIDR in 2006, and it became part of both of them. So what we have actually is a microbial repository of about 20,000 isolates. It's a comprehensive collection spanning, in Lebanon and wider region. Also, we have done 25 plus regional trainings in EMR and AFRO. Now, what we do actually, as I mentioned, bacterial pathogen surveillance, outbreak response, AMR monitoring, and capacity building in the regions. And the core capabilities in the center is doing next generation sequencing, regional bacterial pathogen repository that I have mentioned. And the repository coverage has a number of bacterial pathogens that are listed. Now, the core capabilities and regional impact is through phenotypic microbiology, genomic epidemiology, and capacity building. And this is bridging lab excellence and public health action. Now, most importantly is the outbreak alert and response, whereby any time that we have any kind of an outbreak that occurs in any country in the region, the WHO requests that we do, the testing. Like more recently, they called for meningitis and in Syria, but actually so far the results were negative. Now also the most important thing is the cholera outbreak response that we have done. So WHO actually nominated the center as a national testing center for cholera, whereby all the isolates, the samples that were clinically suspected, they were confirmed in the lab by phenotypic testing as well as by molecular testing. Now also the global and regional network, there is an integration of GORN, of GTFCC, WHO Alliance, and PulseNet. Now the equipment that we have include a number of equipment from real-time PCR to PCR to others, qubit, 4 fluorometers. And we have more recently placed equipment like the Vitek MS Prime, and this has been facilitated by Dr. Antoine Abou Fayyad. And the second one is the Vitex of bioMérieux. And we were able to purchase through the CDC CRDF grant the Illumina MySeq I100 Plus. And also we have them in ion devices that were purchased on grants. Now, in addition, and more recently, there was an initiative by Dr. Melhem Bilain, who applied for a grant that will, build capacities in the middle-income countries. And this grant actually provided shipment of instruments up to the value is up to $900,000. And in this respect, I would like to thank Dr. Sawaya, the Dean and VP of the Faculty of Medicine, who supports us by providing funding in order for this process to occur and be implemented. Now, the strategic impact and future directions, we need to expand the repository, support Lebanon National Cholera Surveillance, and strengthen GORN and PulseNet partnerships. And this is a previous, picture of the members that constitute the WHO Collaborating Center from EPIM and CIDR. And thank you. Now we will start the session of the different lectures, starting with the basic sciences and with Dr. Michel Masad. Dr. Michel Masad is an associate professor at the Department of Experimental Pathology, Immunology, and Microbiology.
Good morning, everyone, and thank you for coming early on Saturday morning. Most of you know my lab's work on inborn errors of immunity, but when SARS-CoV-2 appeared, we decided to study the immune response to SARS-CoV-2 infection, starting initially with looking at patients with severe versus mild course of infection. And then we had a longitudinal study on the immune response to SARS-CoV-2 infection in the pediatric population. And then when Lebanon decided to import the Pfizer mRNA-based vaccine, we started the study that I'm going to talk about, which is on the longitudinal T and B cell actual recall responses following 2 doses of vaccination with this vaccine. So SARS-CoV-2 is a enveloped positive strand RNA virus that first appeared in Wuhan in 2019, and it was associated with a severe respiratory disease called COVID-19. And then SARS-CoV-2 rapidly spread out of China to affect the world and became a pandemic. And then after almost 6 years and multiple mutant strains, it has already infected over 800 million individuals and caused the death of more than 7 million patients. So the spike protein here is the surface protein that mediates the infectivity of SARS-CoV-2 by binding to the ACE2 receptor on the epithelial surface of the mammalian cells. And this mediates the internalization of either the full virus or fusion of the virus with the plasma membrane to inject onto only its RNA. And once the RNA is inside, it's translated in the host ribosome to make the viral proteins, make new viruses that will exit from the cells after lysing them. The SARS-CoV-2 has been associated with a lot of clinical manifestation ranging from very mild to severe. But one of the most severe manifestation was immune mediated. Basically, in some of the most severely infected patients, they had production of a high level of pro-inflammatory cytokines that caused a cytokine storm, resulting in multi-organ damage and eventually increasing highly the incidence of death among these patients. Because of the importance, infectivity, the lethal outcome of SARS-CoV-2 and the pandemic nature, a lot of companies start making vaccines against it. Today, I'm going to talk only about the RNA-based vaccine that was spearheaded by Moderna and Pfizer-BioNTech. This vaccine actually is an RNA molecule that has been modified to increase its stability and longevity. It encodes the spike protein. It's encapsulated in the lipid nanoparticle. And when it's given to the recipient in the shoulder muscles, the lipid nanoparticle fuse with the plasma membrane, inject the RNA into the muscle cells, and then those muscle cells now start making the full-length spike protein, which migrate in the lymphatic system to the local draining lymph nodes to activate B cells. But also this protein is picked up by dendritic cells that chop it into small molecules presented on MHC on their surface and migrate to the draining lymph nodes to activate T cells. And this is where the adaptive immune response is generated secondary to vaccination. There have been numerous studies on the different aspects of the immune response to vaccination with the Pfizer vaccine, BNT162b2. I'm going to summarize them very briefly and chronologically. The initial studies after vaccination, 1 to 2 weeks after vaccination, involved looking at the production of spike-specific immunoglobulins. And it was shown that 2 to 4 weeks after vaccination, the recipients developed high levels of IgG and IgA against the spike protein. Then people introduced neutralization assay to study the neutralization activity of these immunoglobulins, and they were highly neutralizing. Then people studied longitudinally the humoral response against these after vaccination, and it was demonstrated that the antibodies last long and they have neutralization activity against the original strain and to different levels against the mutant strain. This is the humoral response, antibody-mediated. There were also studies on the B-cell immune response, and it was demonstrated that after vaccination, there are S-specific, S stands for spike, spike-specific memory B cells that develop 1 month after the second dose, and they were detectable for 6 months. Plasmablasts, which are the factory of antibody production, develop in the blood 1 week after the second dose, but then decline to become undetectable in the blood, but they were still detectable in the lymph nodes up to 6 months after immunization. And it was concluded at that point that immunization with this vaccine provided memory, long-lasting memory, germinal center reactions, and B cells, plasmablasts, that can secrete antibodies and provide protection for up to 6 months after immunization. T cells were also studied, and after vaccination, it was demonstrated that there was development of spike-specific memory T cells as early as 1 week after the first dose, peaked 1 to 3 weeks after the second dose, and were detectable for more than 8 months. Most of these cells were of the memory phenotype, and they had a Th1 polymerization phenotype, which is important because they mediate viral immunity with very little involvement of Th2 and Th17, which is also important because you don't want a vaccine to cause inflammation. So the vaccine was doing what is expected out of it. T follicular helper cells, which are the drivers of plasmablast generation, the rare cells in the blood, were also detected in the blood, but in very low levels and disappeared very quickly. However, they were found in the lymph nodes up to 6 months after vaccination, which shows that they go to the lymph node to mediate plasmablast differentiation and secretion of antibodies. So all of these studies were really very comprehensive. However, they had some limitations. In most of the studies, the B cell and the T cell immune responses were analyzed separately. The analysis was initially short, and long-term studies relied on random sampling from a large number of individuals. And when you randomly sample people, one at 6 months, one at 2 months, you get different levels of antibody production or B-cell development, which might not accurately reflect the true kinetics of the immune response within the same individuals. Each of us has a different immune response, so I could make a strong immune response at 2 months, or you can make a weak response at 6 months. So it's not really very kinetic. Many of the studies later included a mixture of infected and vaccinated individuals, somebody infected then vaccinated once or twice, or vaccinated followed by an infection. The recall response, like the ability of the B cells to secrete antibodies, was never investigated. And the T-cell responses relied on very short-term assays of 1 day, which is the stimulation of the T-cells with peptide-bound tetramers, and tetramers mimic MHC molecules, but they are of limited diversity, and they are bound to only a single peptide. And this allows the detection of HLA and epitope-restricted S-specific T cells, which is not predictive of the full spectrum of the recall response. When we conceived the study in 2020, and even before we knew what other people were doing, we designed it to be longitudinal by nature, with repeated sampling from the same individuals for at least 9 months after the second dose. This will allow us to study the humoral response within the same individuals up to 9 months. We wanted to do a detailed immunophenotype of the B cells longitudinally, but we also wanted to stimulate the B cells exogenously, ex vivo, to induce the S-specific immunoglobulin secretion. Most of these studies, as I said, studied the T cells very shortly for 1 day. We wanted to induce the proliferation of T cells because the first response T cells do after activation with an antigen is to proliferate. So we wanted to induce their proliferation with the spike protein and then study their immune phenotype longitudinally. And by doing this, we thought we would allow to perform correlation studies between all branches of the adaptive immunity because all of them were done in the same study. So the study design was to recruit 12 individuals and obtain 7 or 6 blood samples from them, 1 before immunization, 1 21 days after the first dose, then 1, 3, 6, and 9 months after the second dose. And at 6 months, 1 individual dropped out. And at 9 months, 2 individuals got infected with COVID, and they agreed to give another sample 1 month later. And another person stayed for 12 months, it's not shown here, and then got a 3rd dose of immunization and agreed to give 1 dose later. So we wanted to include them in the study. So we obtained the samples from these individuals, and we isolated the plasma and the PBMCs. The plasma was used in an in-house ELISA using the S protein, full-length S protein, as well as the RBD domain as antigen to determine the levels of IgM, IgA, and IgG against the S protein and the RBD domain. We also used the plasma to do a neutralization assay to prevent the infection of Vero E6 cells with live SARS-CoV-2 virus. The PBMCs were split into 2 portions. 1 portion, we did a detailed immune phenotyping of their B cells, and then we stimulated their B cells 14 days with anti-CD40 and IL-21 to induce IgG secretion. And then we checked the level of IgG secreted by ELISA and the neutralization potential. For the T cells, as I said, we stimulate them for 9 days with the spike protein with anti-CD28 and IL-2. And then we gated on the spike-specific cells that proliferated and did a detailed immune phenotype of their nature. So I'm going to start with the humoral immune response. In H1, the top is this is an ELISA of the spike protein to detect IgM in the top, IgA in the middle, and IgG in the bottom. Each line is 1 individual, and in red is the average of all the individuals. And this data demonstrated that the level of spike-specific IgM peaked 21 days after the first dose at around 6-fold increase. IgA and IgG against the spike protein peak 1 month after the second dose at 20 and 30-fold increase respectively. But more importantly, even if these decrease with time, their levels stay above the background or above pre-immunization levels, which demonstrate that this can last up to 9 months after immunization. And then we checked the level of RBD-specific immunoglobulins. If you focus on the last panel here, starting with IgM, in gray is the level of IgM against the RBD peptides, and in black is the one against the spike peptide that I just showed you. And you can see that IgM also peaks at 21 days. IgM against the RBD peaks at 21 days, and then follows a similar kinetics to that of the anti-spike immunoglobulin. IgA actually peaks at 3 months after the second dose, which is later than the spike protein, and then starts decreasing. And this indicates its sustainability of the RBD-specific immunoglobulin in the serum of the individuals and disappearance of the other type of spike-specific immunoglobulins. IgG appeared 1 month after the second dose, but it was stable throughout the study, whereas the spike-specific immunoglobulin decreased. So to determine the function of this immunoglobulin, we introduced a neutralization assay in the lab where we infect VIRO-E6 cells with SARS-CoV-2 virus. And then we checked the ability of the serum isolated from all the donors to neutralize the virus and prevent its affectability. And all of these are the serial dilution of all the sera at the different months. And the summary of the study is here. This showed that the serum that was obtained 3 months after the immunization had the highest IC50 and the strongest neutralization potential, which actually correlated with the highest level of IgA and IgG against the RBD domain in the serum of the individual. So as I said, two people got infected with SARS-CoV-2 9 months after the second dose, and 1 person obtained the third vaccine 1 month after the 12 months after the first dose. So we obtained samples from them, and then we checked their level of spike-specific IgM, IgA, and IgG, as well as RBD-specific immunoglobulin. And as you can see, we observed a surge in the level of immunoglobulin secreted after infection or a third dose of immunization, demonstrating that the vaccine can initiate the immune response against further infection or immunization. And importantly, this after infection or immunization, the immunoglobulins generated were highly neutralizing, much more than what was observed at the peak values for these individuals. So then we wanted to look at the PBMCs, B cells, and T cells. Started with the B cells. This was our gating strategy. We obtained the PBMCs from the donors. We gate on the peripheral PBMCs. We eliminate doublets and dead cells, and then we stain the B cells with CD19. Then we co-stain them with CD19 and the spike protein to identify the spike-specific B cells, and we determine the percentage of CD27 positive memory B cells. And then of those that switched from IgD to another isotype, we determined the percentage of the IgG positive and IgA positive spike-specific memory B cells. So we did that on all the samples, and we found out that the peak percentage of spike-specific B cells is at 1 month after the second dose. It peaks at around 44% of all the total B cells. The percentage of memory B cells keeps increasing with time until the 9 month, while the percentage of naive spike-specific B cell decrease. And of those spike-specific B cells, the percentage of IgG positive keep increasing with time until the 6-month time point. And similarly, the percentage of IgA positive B cells peak at 3 months after the second dose and stays up to 9 months. So this demonstrated that immunization can result in the production of memory B cells, that is of IgG and IgA type, that can last up to 9 months and maybe longer. So as I said, we also stimulated these B cells with anti-CD40 and IL-21 to induce IgG secretion. And we checked in the top the levels of S-specific immunoglobulin secreted, and in the bottom RBD-specific IgG secreted. And we found that the level of antibodies secreted by these B cells peaked 6 months after the second dose, which was correlated with the level of memory B cells identified by flow cytometry. And importantly, all of these secreted antibodies are very neutralizing, and the peak neutralization potential is 6 months after the second dose, which is different than what have been observed in the plasma of these individuals, because in the plasma, the B cells are not re-stimulated. Whereas here, we actually took the B cells from the patients and re-stimulated them and showed that there is enrichment of memory B cells against the spike protein, which result in more antibody production later on. And these two individuals who got infected and the person who got the third dose of vaccine had a resurge in the percentage of spike-specific B cells, more memory B cells made, and additional IgG switch and IgGA switch memory B cells after the infection or immunization. They secrete S-specific IgG and RBD-specific IgG that have a very high neutralizing potential. So 2 doses of vaccination followed by either infection or 1 extra dose of vaccination gives the B cells the ability to re-amplify and secrete highly neutralizing antibodies. So we also looked at the D-cell immunophenotyping of these patients. And as I said, we actually activated the T cells before because we want to induce their proliferation, but we used the S antigen, S protein as an antigen to induce the proliferation of only S-specific T cells. And this is our gating strategy. We gate on the proliferated cells, we eliminate doublets and dead cells. Then we look at CD4 positive cells that were able to dilute CFSE, which is a proliferation dye. And these are the cells that proliferated when stimulated with the S protein, which means that they are antigen specific. Then we study their naive memory phenotype, the percentage of T follicular helper cells, and their activation markers, CD25 and HLA-DR. These are surface protein. Intracellular, we look at their ability to produce interferon gamma and IL-17 as markers of Th1 and Th17 differentiation. We did the same study for CD8 cells. We gated on the proliferated CD8 cells. We looked at their naive and memory phenotype. And then intracellularly, we looked at the level of interferon gamma production, which is a pro-inflammatory cytokines. But also we looked at their levels of intracellular granzyme and perforin, and these are the mediator of CD8 T cell lysis. So the higher granzyme and perforin there is, the more lytic the T cells are. And this is what you want your CD8 cells to be after immunization. So we did the analysis of all the samples, and this is looking at CD4 T cell immune phenotyping. These are the total proliferated CD4 T cells. You can see that the CD4 cells actually peak of proliferation was around 21 days after the first dose, and it stayed stable up to 9 months during the study. Most of these T cells were of memory phenotype. Importantly, their level of activation was at the highest peak 21 days after the first dose, which means that they are activated, but they don't maintain their activation for a very long time. You don't want the T cells to be activated continuously after vaccination. Most of them were of the Th1 phenotype, which is important for the fight against viral infection, and that peaked at 1 month after. So they polarized to Th1 with minimum polarization to Th17, which means that they can fight infections, but they're not pro-inflammatory. And remember when I said that T follicular helper cells were rare in the periphery as detected by other people, and they were mostly in the lymph nodes? Because we induced their proliferation, we were able to detect the T follicular helper cells appear 21 days after the first dose, and then go down and disappear by 3 months after the second dose. And these T follicular helps are important in the periphery because anytime there is a new virus coming, they go to the lymph nodes, and they induce new round of B cell activation and immunoglobulin production. CD8 cells were also immune phenotypes, and contrary to CD4, CD8 peaked 1 month after the second dose, but I stayed up through the study. They were mostly of the memory phenotype with enrichment of this red line, which are the effector memory cells that are in the periphery, also ready to fight viral infections. They don't produce a lot of interferon gamma, and this goes down with time, but they produce a lot of perforin and granzyme, mainly 21 days after the first dose that stays up, and this is the window of protection that they provide after vaccination until they are re-exposed to a different infection. So because we did all of these extended B and T cell studies in the same work on the same samples, we were able to do very strong correlation studies. And I'm going to summarize this to you. This seems overwhelming, but I'm going to summarize it very quickly. We found a very strong and significant correlation between the percentage of IgG positive memory B cells and both the plasma levels and the supernatant levels of anti-S and anti-RBD IgGs and their neutralization potential. And there is a good correlation between those B cells and the antibody they produce and the function of those antibodies. We also found a strong correlation between the level of IgA positive B cells and the level of IgA in the serum and the plasma and their neutralization potential. We also did some correlation studies between CD4 cells and B cells, because T effector CD4 cells are the master regulator of the immune response. And we found a good correlation between the memory T cells, CD4, and the memory B cells, IgG and IgA switch, as well as with the antibodies they secrete and neutralization potential. And we found a very strong correlation between CD4 T cells and the Th1 phenotype, which induce viral infection, and the levels of perforin and granzyme in CD8 cells, which solidify CD4 cells as the master regulator of B cells and T cells, and CD8 T cells. So in summary, we did a longitudinal study. We repeated sampling from the same individuals to follow the kinetics. We found basically that CD4 cells are the first to proliferate and increase in number. They polarize to Th1. And by doing so, they induce the activation of CD8 cells. They help them to make more perforin and granzyme that is ready to kill virally infected cells. Some of those Th1 cells become T follicular helper cells, then induce B cells switching to IgG and IgA and help them produce a large amount of neutralizing spike and RBD-specific IgA and IgG to provide protection for at least 9 months, but we have the data that shows more than 12-month protection. At the end, I want to acknowledge the people who did this work. This work was mostly done by Yumna Al-Orfali, who is a PhD student and an RA in the lab. She did most of the experiments with help from a previous postdoc, Rana Mansour. Habib Al-Alamouni was involved in almost every experiment, but he was mostly in charge of the neutralization assay. Ziad Jabour and Antoine Abou Fayyad's lab helped us sequence the viral strain that was isolated. Esber Seba provided us with the RBD peptides to do the ELISAs with, and Ghada Khawaja is a collaborator from Beirut Arab University and a co-PI on Yumna's thesis. And this was funded to different level by the CNRS of Lebanon, by the Medical Practice Plan, and by a Pfizer Independent Research Grant. Thank you very much.
Thank you, Dr. Mas'ad. The questions will be kept to the end of the session. Thank you. The next speaker is Dr. Ilyes Rahal, professor at the Department of Experimental Pathology, Immunology, and Microbiology. He will be talking about the immunomodulatory effects of the EB virus.
Good morning, everyone. I want you to take a second today and take a look to your right and then to your left. Look at the people sitting next to you. Do you know what very likely the person next to you has? That's EBV. It's a very, very common virus. The other thing you might not think about is that, again, if you take a look to your left and to your right, the person sitting next to you might be shedding this virus now because it can reactivate. It's one of these viruses that go into latency. And once you have it for life. You'll never be able to get rid of it. And it's a virus that very commonly reactivates, meaning it re-replicates and you go on and shed it. So while Selena is doing her magic, I think I'll go on. So it's a very, very common virus. More than 90% of us have it. Once you're infected with it, you have it for life, and very frequently reactivates. And what happens then is that you shed its antigens. And one of these antigens is its DNA, which is known to have immunostimulatory properties. So this means that this DNA can stimulate your immune system and do things with it. So the virus itself is associated with various diseases. The main one it's known for is infectious mononucleosis, which is a flu-like entity. It's mild to an extent. And in addition to this, the virus is oncogenic. So this means that some people might develop cancers because of having this virus. It has oncogenic properties. And like I said earlier, and this is the interest of the lab, that this virus is immunostimulatory, not just its DNA, but I'll be focusing in the early part of my talk today on its DNA. All right, so knowing that this virus is associated with so many different types of autoimmune diseases and pro-inflammatory diseases, we started thinking early on, like about 10 years ago, that could it be that there's something that unifies the inflammation that happens, so that it can be associated with all of these different diseases? Is there something that kind of helps the milieu and the infected person to be pro-inflammatory in such a way that you develop autoimmunity or so that you develop inflammation or autoimmunity. So we started taking a look at one specific antigen that might help things out. I'm not saying that this is the autoantigen. It's something that just helps out inflammation. We thought maybe it could be the DNA. So we took the DNA, we gave it to mice. That's about 10 years ago. We gave it to mice. We looked at what's going on in the mice, and we noticed something, that there's increase in pro-inflammatory mediators such as interferon gamma, but something that interested us was interleukin-17. We saw that it increases in mice once you give the DNA. So what is interleukin-17? I think Dr. Masad kind of paved the way. He mentioned this a little bit earlier. It's a pro-inflammatory cytokine. It has a role in combating infectious agents, but it's also known to be associated with autoimmunity. This is why we're interested in this. So we examined as well the transcription networks that are induced by the DNA. So we used RNA sequencing. We incubated basically mouse immune cells, peripheral immune cells, and we incubated that with the DNA. We examined what transcription networks existed. We saw an increase in lots of pro-inflammatory cytokines. The usual suspects here, the top ones, were interleukin-17 or interferon gamma, but also importantly, toll-like receptors, endosomal toll-like receptors. I'll go back to this in a little bit. So we saw this interesting network. At the center of it was interferon gamma and interleukin-17, as we had kind of expected. Examining what pathways had been enriched using gene enrichment analysis, gene ontology networks showed us that there is enhancement of inflammatory pathways such as interleukin-17 production. In addition to that, the KEG pathways, which sometimes show you some of the diseases. So basically what it shows you is the genes that are expressed, enhanced in their expression in certain diseases, such as inflammatory bowel disease, rheumatoid arthritis, type 1 diabetes. In addition to that, we also saw this again, which is toll-like receptor pathways. We saw enhancement in the expression of their genes. So all of this pointed towards toll-like receptors as a potential mediator that is detecting this DNA and resulting in expression. So the paradigm in immunology is that you have your antigen. It's detected by an antigen-presenting cell, usually a professional one, and then resulting in presentation to T cells and production of cytokines. So we wondered what kind of antigen-presenting cells were playing a role in this, and what kind of receptors were playing a role in this pathway, resulting in enhancement of interleukin-17. So to make a long story short, there were different indicators, but we focused on endosomal toll-like receptors. These are ones that can detect nucleic acids. So we saw that if we take the blood cells, the immune cells from mice, the PBMCs, and we give them the DNA, we have enhancement of interleukin-17 in culture, also in mice. But then if you inhibit toll-like receptor 3 or 7 or 9, which are endosomal toll-like receptors, you see a decrease in the production of interleukin-17. So this kind of tells us that it's probably these receptors that are detecting the DNA or mediating at least this pathway. So this happens ex vivo in culture as well as in mice in vivo. So again, going back to that paradigm, what antigen-presenting cells could be playing this? The main professional ones are the B cells, the macrophages, and the dendritic cells. Again, to make a long story short, we did co-culture experiments putting different types of cells together, and the best expression or production of interleukin-17 was when we co-cultured dendritic cells with the T cells and the EBV DNA. So this tells us that probably the professional antigen-presenting cell playing a major role in this pathway is the dendritic cell. Now, you might be wondering, so I'm talking a lot about the DNA. What about the virus? Does it do something similar? And indeed, we saw that if we take the virus, we do similar co-culture experiments, we do see, again, dendritic cells, T cells, and EBV variants, the intact viral particles. This results in enhancement of production of interleukin 17. All right, so this is all in mice and in culture in the lab. What about in real life? Does it matter in humans? So in order to test whether this plays a role in an actual disease, we examined rheumatoid arthritis subjects, and in them we saw increased numbers of EBV DNA copies in their blood, in addition to an increase in interleukin 17A in their sera. But more importantly, we saw a positive correlation. So the higher the amount of DNA you have in the blood, the more you have interleukin 17. We saw this in the RA patients, but not in RA controls. So it looks like this pathway also is important in humans. This increase caused by the DNA in interleukin 17, among other pro-inflammatory mediators. Now, from there on, the lab we started using different animal models to kind of replicate circumstances in the lab and ask different questions. There's a lot of storylines in different diseases we look at the lab, which is inflammatory bowel disease, diabetes mellitus, but I'll focus today only one storyline, which is that of rheumatoid arthritis. So we used the chicken collagen type 2 model here. What you do is you take your mouse, you give it the chicken collagen, and then you expect to see arthritis. It's a very well-validated model. The nice thing about this model is that once you give the instigator your chicken collagen type 2, only 40 to 60% of the mice are supposed to have symptoms. So it's relatively a mild arthritis. So this allows us a margin to see an increment when we give the virus or its DNA. So first, we examined whether giving the DNA would enhance the incidence of arthritis, and indeed, we saw an enhancement of arthritis in these mice. We also saw different indicators of disease, such as the height, paw thickness. It increased significantly, among other things. But quite importantly is the activity index or the clinical score of this disease. And we saw that giving the DNA enhanced the clinical score in these mice, as well as tissue destruction and so on and so forth. So giving the DNA enhances arthritis in mice. Now, as you probably know, nowadays everybody thinks about the gut microbiota and our microbiome, and I think you'll hear more about the microbiome later on with Dr. Bilain following me, I think. So one axis here that we know about is that the microbiome is shaped by inflammation, and inflammation shapes the microbiome. So we wondered whether giving the DNA results in enhanced inflammation. This shapes the microbiome in such a way that it also exacerbates inflammation. So we examined the stool microbiota, and we saw dysbiosis, and this means there is a change in the phyla and the genera that you see in these in the gut of these mice, and it was in a pro-inflammatory manner, as you will see on the next slide. Importantly, we saw a change in the richness and the diversity, as well as a decrease in rare numbers or ones that are rare in the gut microbiome. So there's a change, and this change also was reflected at the level of the different organisms. So if you take a look at the relative abundance of various genera, we saw changes at about 30 genera that were relevant in an inflammatory manner. Just give you a couple of examples. So for example, Desulfovibrio. So this organism, the more it is there in the gut, the more you expect to see inflammation. And if you compare the DNA administered arthritis mice to the simply arthritic mice, you'll see an increase in the relative abundance of these organisms. In addition, we saw a trend of decrease, for example, in Lactobacillus. Lactobacillus is known to be anti-inflammatory, and it's a probiotic that people sometimes use. So we saw a decrease. Have an issue?
Sorry.
So I guess for the sake of time, I'll go on as they're doing this. So In addition to that, we wondered whether the change in the microbiome within these mice, if that by itself is sufficient to enhance arthritis. So forget about the DNA, but whatever has been changed in the mice. So for this purpose, what we did was a fecal microbiota transplantation or fecal transplantation. And what we did was take I'm good. No, I'm fine. Go ahead, do whatever you want. All right, so we took the feces from the mice that had been given the DNA. We took these feces and gave them to naive mice that were not exposed to the DNA. And indeed, what we saw was an increase in the incidence of arthritis again, an enhancement in the clinical features. And so this basically tells us that the DNA by itself is sufficient, or excuse me, the feces, the change in the microbiome by itself is sufficient to induce arthritis in mice. So it's the microbiome that has been changed that is also sufficient to do this. So to make basically things, just summarize things for what I've said to you guys today. We believe that an EBV infection will result in shedding of the DNA. It looks like that's the major antigen to do this. We think that, well, based on our data, that toll-like receptors 3, 7, and 9, these are the endosomal toll-like receptors. They will sense this DNA, and this will result in enhancement of production of interleukin-17, among other mediators that we've looked at, and we see enhancement of inflammation and disease exacerbation. All right, so finally, what I want to do is thank all the people in the lab, the current members, of course, but I want to thank specifically the people whose work was featured today, Dr. Helen Hajjar, who basically was the first person to show us that the DNA enhances interleukin-17. Sarah Jishi, who looked at the various endosomal toll-like receptors. It's her and Dr. Marwa Sheb, who basically figured out this pathway that I just showed you. The work on arthritis was conducted by Dr. Sokaina Fadlallah and Dr. Noor Shirri. They figured out what's going on with the microbiome, and Noor also worked on something that I didn't really talk a lot about today, which is the fact that we gave endosomal toll-like receptor inhibitors to arthritic mice, and that was good in making these mice feel better. So it looks like what's happening with the arthritic mice is also mediated by the endosomal toll-like receptors. And finally, 3 research assistants, former research assistants who are now PhD students, Noor Saloom, Hadi Hussain, and Aya Qasim, who had their basically their hands in the different pieces of the pie, and without them, this couldn't have moved forward. I'd also like to thank all my collaborators and the various funding agencies who helped us with this work. All right, thank you.
Thank you, Dr. Rahab. The next speaker is Dr. Melhem Bilain, Assistant Professor at the Department of Experimental Pathology, Immunology, and Microbiology, talking on the human gut microbiota.
All right. Hello, everyone, and thank you for your time on Saturday to come and hear about our progress in the department. Today, I'll be presenting about our recent findings in a study that I conducted in collaboration with Dr. Fadi Murad at AUB-MC on ulcerative colitis patients. So in brief, ulcerative colitis is a chronic relapsing inflammation of the colon. And it is usually known, the known symptoms of such disease is the abdominal pain, diarrhea, rectal bleeding, weight loss, fatigue. Now, the major type of treatment for ulcerative colitis is the immune suppression. But every time, not every time, but in most of the cases, in severe cases as well, every time we give anti-inflammatory guided drugs, it ends up by relapsing. So we move to other type of drugs like mesalamine, steroids, and even more expensive ones like biologics. But the big question is, why do relapses persist? And we already think that we have the optimal therapy. So This gives us a room to think a bit larger. Now, what if part of the explanation and part of the solution lies in the gut microbiota? We usually tend to study the bacteria as pathogen, but we should start thinking of these organisms as potential treatment. Now, why do UC matters and why the gut microbiota matters in the UC? The UC mainly is not only an inflammatory disease of the colon. It also occurs in the body part that is the most densely colonized by bacteria, and this is the gut microbiota. A healthy gut microbiota, meaning that a region that is known to be colonized by a specific relative abundance of organisms in a healthy way, is known to contribute to epithelial integrity, immune tolerance, anti-inflammatory signaling, and so on. Once we have an imbalance, meaning that certain organisms overgrow other organisms, or certain organisms get completely depleted or absent in our gut, we start seeing diseases. And this has been shown in plenty of papers. If you go on Google or PubMed, you put gut microbiota and whatever you put, a disease, you will find a paper reporting that there is an imbalance in the gut microbiota there. And let's say psychiatric disorders and cardiovascular diseases, even and skin as well. So unpredictable, we don't know what it does, but it is correlated everywhere. Most of the data in ulcerative colitis has been published in Western population. And there is an expectation that ulcerative colitis will be increasing by 2030, at least 20 times more than what we have here. But the most important factor is that when we try to screen for ulcerative colitis or diagnose ulcerative colitis in African population, mainly in hunter-gatherer tribes, we don't see it. So if we go to the Hadza population or the Pygmy population, who are the hunter-gatherers, there is zero incident of ulcerative colitis. Is it because they lack our diet or our industrialized environment when it comes to diet, to habits, to the environment, to the medicine, and so on? Does this really impact their gut microbiota? Do they have a different gut microbiota? So this was a work that I've done previously, and yes, they have a different gut microbiota than ours, and we are relying on their bacterial composition there to say that this is the ancestral gut microbiota, and with our modern lifestyle, we have impacted it that way, and this might be leading to the disruption of the balanced community there. What we did at AUB and AUB-MC, we collected fecal material from ulcerative colitis patients and from healthy donors, of course, after the IRB approval. We faced a bit of hard time collecting samples and convincing patients to give samples, but this is a point here that I would like to emphasize, that we really need to collect specimen from patients who are either ulcerative colitis or other things. I have many ongoing projects, but we need to encourage the patient to give us these samples so we can study it and generate our own data. Because the gut microbiota is not only impacted by diet, medicine, and so on, but also by the where we live. So the Lebanese microbiota, gut microbiota, or other microbiotas is different than the ones in France and Germany and so on. So we were able to secure 11 ulcerative colitis samples and 11 healthy samples. So we conducted a pilot study. It was the first in Lebanon to do this, and we wanted to see molecularly what's the composition of the gut microbiota in these patients. So we first started by sequencing the fecal material and sequencing the V3, V4 region of the 16S sequence, which is like a region in the DNA of any bacteria that allows us to identify it at the species level. And we performed the sequencing using the Illumina MiSeq i100 platform. After bioinformatic analysis, we were able to see that there was a significant reduction in alpha diversity in ulcerative colitis patients in Lebanon. This means that you have a selective loss of evolutionary distinct taxa. And at the same time, we were able to show that the beta diversity in the UC patient harbors distinct gut microbial community. And the dysbiosis of the gut microbiota of UC patients in Lebanon is mainly driven by chronic mucosal inflammation, epithelial barrier, as well as environmental factors, technically the diet and the medication. So the key message here is that the UC in this population represents a targeted loss of protective microbial functions, not a uniform depletion. Moving on to a deeper taxonomic classification of the organisms that we saw in the gut microbiota of the UC and the healthy donors. There were dominant phyla, which is similar between the UC and the healthy one and the healthy donors. They were mainly dominated by Bacillota, Bacteroidota, and Actinomycetota. And there were proteobacteria that were remarkably low in ulcerative colitis patients, unlike the reported UC cohorts worldwide. At the same time, there were three major genera, the Ruminococcus bacteroides and Coprococcus. These all are major short-chain fatty acid producers that were depleted in the gut microbiota of all 11 UC patients. And these bacteria are known to produce short-chain fatty acids, which are metabolites that prevent inflammation or reduces inflammation by their capacity to inhibit inflammatory pathways. So the functional implication of these changes means that we have a reduced carbohydrate fermentation in ulcerative colitis patients. We have a decreased in acetate, butyrate, and propionate availability, which are all anti-inflammatory metabolites. And therefore, we have an impaired epithelial barrier integrity and anti-inflammatory signaling in UC patients. Now, the take-home message from this results is that the UC cohort they are marked by a loss of protective microbial functions, not only expansion of classical pathogens. Now, the most important result, which was, I think, somehow a breakthrough, because all ulcerative colitis patients in the world and all the metagenomic studies that have been done on stool samples collected from ulcerative colitis patients, they showed a depletion of a bacteria that is called Faecalibacterium prosnitzi. And Faecalibacterium prosnitzi is known to be one of the bugs that produce major anti-inflammatory short-chain fatty acids. In our study, we found that Faecalibacterium prosnitzi was unchanged in all 11 UC samples that we collected. So the first idea was that we had a problem in the sequencing. We have to resequence it because almost 80% of the startups now trying to cure ulcerative colitis by microbial communities, they are relying on the presence or absence of Faecalibacterium prosnitzi. So I shipped the samples to Korea and I had it resequenced there to make sure that there was no errors at our end in our platform. And the results came similar to our results. So this is a first of a kind where we see that the major bacteria known to be depleted in ulcerative colitis patients, and most of the companies are trying to build their treatment, bacteriotherapeutic treatment around this bug, is overrepresented in our population. Now, is this due to our diet? Is this due to other bacteria colonizing and permitting this bug to be present, or this opens up a new era of bacteriotherapeutic potential of the gut microbiota in the Lebanese population, where we can try to identify organisms that we disregarded it, but might be major players in ulcerative colitis. So what does this mean? The first thing is that ulcerative colitis is not only immune mediated, it's a disease of microbial ecosystem disruption. And also, dysbiosis of the gut microbiota contributes to the disease persistence and relapse. And the solution, or part of the solution, is restoring the gut microbiota composition, because this will be a therapeutic strategy that will allow us to develop a bacteriotherapeutic approach to treat ulcerative colitis. Now, what we can do in this field later on, or it's currently an ongoing process, is Now, applying something called culturomics on the stool samples, because when you sequence the stool samples, you get a bunch of DNA. We attribute to bacterial organisms. We say that this has been overrepresented, this has been depleted. Okay, this is a great Excel sheet that we obtain. We can make beautiful bars with it. But when it comes to creating a therapeutic product, we have to take a step further, which is isolating the biological organisms, the ones that are contributing to the development of the disease, and the ones that are known to be depleted in the gut microbiota of the UC patients. And then know how we can, and then understand how these organisms function, and try to create a safe module that is an alternative to the fecal microbiota transplantation, which is the transplantation of fecal material to patients, and in an indirect way, transferring an uncontrolled environment that might contain multi-drug resistant bacteria that are not detected by sequencing, toxins, and so on. So at this stage, we are making a synthetic microbial community from the healthy donors, and we are making a synthetic microbial community from the ulcerative colitis patients. We are doing mice experiments that will allow us to monitor UC with this community versus the other one, to be able to refine a pill that contains the right bugs to be given for the UC patients to rebalance their gut microbiota. Now, in conclusion, we are trained to eliminate bacteria, and this is highly important because antibiotics usually saves life. But which bacteria to preserve, restore, or even prescribe, this is the major question, and this is gonna be, I believe, the next 10 years research in multiple diseases. And UC and across neurology, cardiovascular disease, and antimicrobial resistance, the gut microbiota is emerging not just as marker of disease, but as a therapeutic target. Now, the question is no longer whether bacteria matter, but how we can safely turn them into medicine. I'm going to just be briefly mentioning the ongoing projects in collaboration with AUB-MC physicians who have been very supportive and very open about studying the gut microbiota in their fields. I'm currently working on the respiratory tract microbiota in electric cigarette smokers versus non-smokers. And the idea is to see the microbial shifts and try to detect biomarkers that e-cigarette smokers might be having a lot at a high level and can be indirectly correlated to certain diseases. We are studying the gut microbiota and alteration in ALS patients. This is in collaboration with Dr. Hassan Damyati. Along with a collaboration with Dr. Ali Tahir, we are also trying to study the microbial frontiers in thalassemia patients. All these have been, we're not reinventing the wheel. All these have been published that the gut microbiota is disrupted. My job is to be able to find the right way to rebalance the gut microbiota of these patients and reverse the symptoms. At the same time, with Dr. Samia Khouri, we are working on gut microbiota and multiple sclerosis. With Dr. Maya Bizri and Dr. Farid, we are working on gut microbiota and psychiatric disorders. And we are working on skin microbiota and skin disorders with Dr. Mazen Korban. Finally, the last two projects are with Dr. Pascal Karam. We are trying to create a synthetic microbial consortium for inborn errors of metabolism. And I talked already about Dr. Fadi's project. And finally, with Dr. Nassim Makaram and Dr. Fuad Zwan, we are running also a big project where we are involving AI to create a microbial-informed nutrition and digital solution for cardiovascular risk reduction. And that's it. Thank you so much.
Thank you, Dr. Wilan. Next speaker, Dr. Esber Saba, Assistant Professor at the Department of Experimental Pathology, Immunology, and Microbiology.
Marhaba. Good morning, everyone. Let me tune my voice a little bit. So today I'm going to present to you a work that we have published recently. It's a collaboration with Dr. Samia Khouri and the MS Center and Khouri Lab. It's about a candidate biomarkers for multiple sclerosis. So we asked ourselves if a biologically meaningful viral immune signal carried on exosomes can serve as a reliable biological marker to predict disease activity and assess future disease risk. Let me start by a small background about multiple sclerosis. So multiple sclerosis is a chronic demyelinating inflammatory disease of the central nervous system and is a leading cause of neurological disability in young adults. There are different forms of multiple sclerosis, with RRMS or relapse-remitting multiple sclerosis being the most frequent. In RRMS, disease activity comes in episodes, whether clinical relapses and or MRI activity like gadolinium-enhancing lesions or new lesions. In multiple sclerosis, this disease activity is highly unpredictable at the individual levels. So our tools are good at assessing the activity after it happens. They are less good at predicting it. And for example, MRI is essential, but it's not always feasible to scan frequently. And even if you manage to do so, subclinical disease activity can still be missed between imaging intervals. On the blood biomarker side, we have neurofilament light chain. This is a very important biomarker to assess axonal damage, but it's downstream, meaning that it reflects the damage that has already occurred. So what do we really need today in MS clinic is a reliable biological markers that is linked to upstream biology that is non-invasive and that is dynamic over time, and a biomarker that is useful for risk stratification in patients. Let me tell you now about two viruses, EBV and HERVs. EBV is one of the most and consistently associated environmental risk factor for MS, with nearly 100% of MS patients being EBV seropositive. But EBV alone doesn't explain how chronic neuroinflammation is sustained in MS patients, which will bring us to the HERVs. These are remnants of previous infection viruses that have integrated our genome long ago. Most are silenced, but some can still be reactivated in certain specific contexts like inflammation. HERV-W, for example, is specifically important for MS because of its envelope protein. I'm denoting it here as p-HERV-W envelope. It's not just a viral marker. This protein can engage innate immune receptors, notably TLR4, to induce neuroinflammation by activating microglia. And also, it impairs remyelination by inhibiting oligodendrocyte precursors differentiation. A key mechanistic link between these two viruses is that EBV can transactivate HERV, providing a plausible bridge between an exogenous trigger and endogenous retroviral inflammatory amplifier. And this synergy is a major driver why we decided to test these two viruses determinants in the same biological compartment that is exosomes. So let me tell you a bit about exosomes. Exosomes are the smallest of extracellular vesicles. They were thought to be the cell's trash bins. But it turns out that exosomes plays a major role in intercellular communication, carrying they are carrying different proteins and nucleic acids, especially microRNAs, to modulate the differentiation or the stimulation of cells at long distances in our bodies. Exosomes are very important for biomarkers discovery because they carry on their surface and in their cargo different proteins that can modulate especially the immune response. But also they are interesting in biomarker discovery in neurological diseases because they can cross the blood-brain barrier. Importantly, in the lab, in Khoury lab, we have optimized a platform to isolate, characterize, and stain exosomes. We've published that exosomes from MS patients carry EBV-related proteins, namely EBNO1, LMP1, LMP2A. And these exosomes can stimulate a pro-inflammatory profile upon treatment on monocyte-derived macrophages. So in this study, we wanted to leverage this platform to ask if these exosomes also carry the p-HERV envelope protein, and if these exosomes can serve as a biomarker for disease activity or to predict long-term disease outcomes. To answer these questions, we used 3 different cohorts. The first cohort is a cross-sectional one with healthy controls, stable RRMS patients, and active RRMS patients. Active MS patients are patients, like I said, with clinical relapse or MRI activity. The second cohort is a longitudinal one with samples collected pre-activity, at activity, and post-activity. And the third cohort here, we wanted to see if the exosomal signature is correlated. It's a retrospective, if the exosomal signature correlated with a long-term clinical outcomes like NIDA or no evidence of disease activity or total number of disease activity events or disability scores. So in the first cohort, we saw that indeed exosomes isolated from active MS patients overexpress both viruses determinants, LMP2A and HERV. And if we look at the correlation between these two markers, we saw that they correlated positively only in active disease, suggesting that these two viruses might co-occur in the same or during inflammatory processes in active MS. In the second cohort, this is where the story becomes clinically relevant. So because we've followed within patients' dynamics of these two markers on the pre-attack, and this is how the staining looks like. So in the pre-attack, at attack, you can see an increased expression. For example, here I'm mentioning HERV. At the attack and post-attack, we see an increase in HERV that peaks around the attack time point. Here we aligned the attack time point to 0, and it decreases later on. And there was a significant trajectory for HERV in the attack for HERV, and this trajectory was not replicated for LMP2A, suggesting that HERV might be a better marker for disease activity as its dynamic followed the trajectory of attack and post-attack. What we've noticed is there was an inverse correlation between the HERV levels at the pre-attack time point and the day to activity. In other words, at the higher the HERV levels at the pre-attack time point, just shorter the time to activity, reflecting that this marker has a potential of predicting disease activity. Then we asked Then we asked whether this upregulation of HERV at the disease activity level, at the disease activity time point, is associated with a broader exosomal marker. So we used the Maxiplex, which is a 37 exosomal markers to phenotype exosomal markers, and we found that there are only two markers that are the CD40 and the CD42A that crossed the false detection rate threshold. The CD40 is the immune markers, and the CD42A is the platelet endothelial marker. When we looked at the correlation structure between the two, we found that HERV correlated with CD40, and the correlation structure suggests that these two markers might be regulated on the same kind of cells. And because of CD40 as a marker for APCs or antigen-presenting cells, this was easy to verify. So we go and stained cells for their expression, stained PBMCs for their expression of HERV, and we found that CD14 that are monocytes and CD19 positive, these are B cells, expressed more HERV than the other cell subtypes like T cells. And when we compare this staining on the cells to the staining on exosomes from the same individuals, we found that both staining correlates positively, suggesting that these cells might be responsible of the pools of exosomes highly expressing HERV in circulation. To lesser extent, B cells, but monocytes are what probably secreting these exosome expressing HERV. In the last study, this is where we wanted to see if high HERV at baseline can predict future disease course. And we used here 25 different patients to phenotype HERV expression on exosomes, and we separated this population based on the median of HERV to create two separate groups as HERV low and HERV high. And in a Cox regression model, we found adjusted for age and sex, we found that high HERV was associated with losing NIDA over the time of the follow-up. Losing NIDA, losing the no evidence of disease activity, that means that losing NIDA is evidence of disease activity with high hazard ratio. And we found that the high HERV group also lost NIDA earlier on average, like you can see here in this Kaplan-Meier image. Also, what we've noticed that high HERV had higher total number of disease activity events, whether clinical relapses or MRI activity. We didn't have any correlation with disease or with the disability scores, mainly because the disability scores reflect more the region of the brain that is affected than better than the subsequent activity or inflammatory activity. Let me end by telling you about what we are doing in terms to follow up this study. We are optimizing ELISA to test for exosomes expression on for HERV expression on exosomes without the need of exosome isolation or the flow cytometry technique. We are doing a high throughput, easier to use test to detect these exosomes expression of HERV. And this test will be implemented. Our preliminary results are very interesting. And this test will be used for a multi-center prospective biomarkers validation study later on. And I would like to thank at the end Dr. Khouri and Khouri lab members, Dr. Mayim Rad and Ms. Layan Naqib, and also thank our collaborators abroad, especially Hervé Perron, for providing the monoclonal antibody and the technical support. Thank you all.
Thank you, Dr. Saba. The next speaker, Dr. Antoine Abou Fayyad, Associate Professor, Department of Experimental Pathology, Immunology, and Microbiology. He will be talking about the Acinetobacter rubble, heavy metals, conflict, and the evolution of extreme drug resistance.
Thank you. Good morning, everyone. Glad to be here and thank you for the opportunity. So I'm going to be talking to you today about, just let me find the pointer first, how conflict can cause pain, suffering, but also resistance that doesn't go away over decades. Basically, in this room here, most of you know that we have a silent pandemic that has been going on for several years now, and it's going to continue if we remain not doing anything about it, which is antimicrobial resistance, and it's claiming too many lives. In 2024 alone, 2 million people died on the planet because of antimicrobial resistance, and we're expecting that by 2050, 10 million people will die every year, which is alone more than any other disease known to man. At the moment with politics, we're all on the news waiting to see if there is a war here or a war there, and there is too many wars happening across. And from also what Dr. Baibo showed, I go a lot to the conflict areas and we see hands-on and on the ground what really happens. The idea is warfare causes widespread infrastructure destruction, and not only medical, but everything. And when you see it and when you experience it, and a lot of us have experienced it here, it becomes even worse to think about other things other than how to survive this. But keep in mind, warfare drives bugs and bacteria in a very weird way, as we're going to see right now. Once this happens, once warfare takes place, it becomes a very strong breeding ground to specific bacteria that thrives. Cholera is one, absolutely, because you have no more wash. But then if you start looking at what happened to the American soldiers when they were being evacuated from Iraq, what happened to what's happening now between Ukraine and Russia, Afghanistan, all these countries, you will hear two words being repeated all the time. Acinetobacter baumannii, Acinetobacter baumannii. And we started wondering why this bug, also known at some point the Iraqi bacter, because in the States back then, Acinetobacter was not a common pathogen you would find in a hospital, right? So we started wondering what makes this bug this prominent whenever you have conflict. And so we started looking at the whole thing from a very wide angle. And whenever we talk about AMR, AMR is due to the misuse and abuse of antibiotics and to the also to the pollution that we cause whenever agro sections like planting or fish farms start using antibiotics in a irrational way. But whenever you look at conflict and AMR and why AMR becomes very aggressive whenever you have conflict. Yes, we have the healthcare collapse. Yes, we have broad antibiotic use and blind antibiotic use. Yes, we have loss of diagnostics because labs usually stop functioning, no more consumables, or lab staff are gone, or other issues. But there is one player that comes in, which is selection and resistance, which we're going to look at in a bit. And selection and resistance comes from pollution, from heavy metals, and from organic compounds that come from rubble and bombing. So the idea is Acinetobacter, as we mentioned, becomes prominent, and clinical chaos comes into place, infrastructure collapses, and one more player comes in as well, which is the population displacement. You have either internally displaced people or people who get displaced from one country to another, which is, so all what I'm saying right now is not a stranger for us. We come from an area where conflict takes place every now and then, unfortunately. So take all of these together and add to it heavy metals, and you have the perfect toxic soup that's going on. And I asked Gemini to, so I told the story to Gemini, which is an AI platform, and I asked him to draw or to visualize the idea. And he drew Acinetobacter growing and thriving with heavy metals around it, making it more and more resistant. So basically, in the soil, we know that because of conflict, you would focus on lead, you would focus on barium, you would focus on cadmium, on antimony, on copper, on mercury, and on arsenic. And these heavy metals specifically, we focus on because we know from several studies that pre-conflict and post-conflict, these are the heavy metals that you will find most in the soil after bombing and shelling takes place, and destruction of buildings as well. So the rubble really makes a perfect place for these bugs to grow. But why? One, your surface area becomes enormous, right? Because you've got rubble, you've got dust, you've got mixed with micronutrients. Whenever we grow bacteria in the lab, what do? We add trace metals, we add micronutrients for the bacteria to grow. This allows bacteria also to form biofilm, and this helps bacteria to focus on gene transfer. So whenever you do conjugation in the lab and you want E. coli to pass on a plasmid to Klebsiella or the other way around, what type of stress do you put on bacteria? Calcium chloride, magnesium chloride, zinc chloride. Why not lead acetate? Have we tried any conjugation with lead? Some people have, and it works. So heavy metals, they do put stress on bacteria and speed up gene transfer and pushes bacteria to pass on plasmids and mobile genetic elements. That's all brilliant. Okay, we're looking at this from the right angle, but one more question was not answered. Why Acinetobacter specifically that thrives in these areas? Two main traits for Acinetobacter that other bacteria don't have. One, genomic plasticity, which allows Acinetobacter, first of all, to acquire mobile genetic elements at the speed of light compared to other bacteria. The second thing is also their ability to acquire genes to deal with heat resistance very quickly. And the second one is desiccation tolerance. The drier the surfaces are, the happier Acinetobacter is. The cleaner the environment is, the happier or the more disinfectant you use, the happier Acinetobacter baumannii is. And these are data from labs. This is data that we've generated and we've seen in the lab. So what did we start doing? Before the war started, around 18 months before the war started, we were doing a One Health AMR surveillance in the country. And you can see on the map, we were collecting from the very north to the very south of the country, water, soil, sewage, wild animals, and other sources as well. And by wild animals, we don't mean any domestic animals. We teamed up with an NGO called Lebanese Wildlife, and they were collecting samples from wild animals for us. We were trying to see how far AMR was spreading in the environment in the country. And our results were a bit worrying. We found quite a lot of clinical samples or samples that we find in the clinics and the hospitals in otters, for example. You know that we have otters in Lebanon. Quite a lot of us were surprised that we have otters in Lebanon, and they are really well protected. But we found E. coli with NDM5 and Acinetobacter in these otters. So this data was not part of AMR and conflict until very recently. And here is why. Also, before the war started, we had a PhD student in the lab from the Beirut Arab University. He works in several hospitals in the south, and he was collecting between the 1st of January 2023 and the end of December 2023 samples from hospitals. We were doing genomic epidemiological surveillance of carbapenem-resistant gram-negative bacteria in the south of the country. And here are the list of the hospitals. They were Sheikh Raghib Harb, Lebanese Italian Hospital, Al-Ra'i, Jabal Amil, Hiram, Hamoud, and Labib. And it was his PhD project to look at what's going on in the hospitals in the south, especially that there is no data published and we don't know what's really going on. But then the war started, and he went back in and he started collecting samples from the same hospitals. So pre-war, he collected 477 samples in 1 year, 365 days. But then during war, or during the escalation of the war, in a period of 66 days, he collected 453 samples. Mind you, 4 of the hospitals were deemed inactive during that period of time. So the period of time is shorter, number of hospitals is less, number of samples is almost as high. Post-war, which is another year, after the ceasefire, so that's November 27, 2024, he collected 665 samples, and all of these are gram-negative bacteria. What did our data show? So we wanted to see, is it really Acinetobacter that plays around and changes the numbers when it comes to this case? And right, we were. If you look at this graph, in the pre-war area, we were collecting per month around 38 samples, and 2 of them were Acinetobacter. During conflict, we were collecting per month 183 samples, of which 30 were Acinetobacter baumannii. Post-war, it doesn't go back straight to pre-war. It takes a while, and we know why, right? So if you look at the percentages, it's about 13 or 14% Acinetobacter baumannii that circulates. And these are the ones that the hospitals diagnose. Remember, during conflict, labs don't operate normally, consumables are not always around, and there is a very, very important player when it comes to Acinetobacter baumannii. Which method do you use to identify Acinetobacter baumannii? 99% of the methods that we apply in the labs are not the recommended ones for Acinetobacter baumannii. So a lot of samples that we receive labeled as E. coli at some point turn out to be Acinetobacter baumannii. And this is common across not only the hospitals in the south, across the globe. The post-war data is very important, and it's very intriguing for us. We thought, why didn't just it drop back? Okay, we know that there is a bit of a latency time at the start. So if you look at the frequency of Acinetobacter per month, it dropped back a bit, and then all the patients who had surgeries because of war injuries, they came back to the hospital. Some of them had infection. That infection was Acinetobacter, war-wounded infection, and then the numbers dropped back. We noticed that based on the hospital data, and this is not our identification, here we know quite well that the identification of the pathogen was a problem, and these numbers will change. And over here, there was an outbreak in one of the hospitals that we were getting samples from. So what did we do? We looked at all the Acinetobacters that were during the war period, and they came from war-wounded patients. And if you look at the middle of this minimum spanning tree, you see that there is a cluster of samples that talk to each other. And this cluster shows these samples, they come from the same hospital. It's not in the same time frame, but they are very much related to each other. And if you look at the peripheries, there are samples within the same time frame in the same hospital, but quite far away from each other. So is it only monoclonal or is it polyclonal? And the observation of Acinetobacter from the hospital in the south also matches what we saw in Libya and what we previously published. We received several samples of Acinetobacter from Libya back in 2020, and we did the same sort of analysis on, and we found that it's polyclonal, the same as what we found here in Lebanon, which also tells you, if you compare an outbreak that can happen here at AUB, and we've published one with Dr. Kanj and the ID team, it's monoclonal. You can have 85 samples, of which maybe 2 are outside this cluster, and everything else falls into the same clone. While whenever you have conflict, polyclonality starts to come. Why? Because 80% of the war-wounded patients who acquire Acinetobacter baumannii, they grab that bug from the environment, not from the hospital. So what we did, we took all the war-wounded patients and we drew a very fancy minimum spanning tree, which personally I'm very proud of. Just to let you know, all of these samples are generated at AUB in-house with our samples. This is around 400 something genomes collected from Lebanon, Niger, Kinshasa, so that's Congo, Syria, and Libya. And we started looking at war-related versus non-war-related. So if you look at the red dots, they are the non-war samples. And if you look at the blue dots, they are the war samples. You remember we said clonal, polyclonal, monoclonal? You can see it at a very big scale. Most of the war-related samples, they don't fall within clusters. You will have a few, obviously. There will be some nosocomial infection or picking up a sample in a hospital, but mostly they are polyclonal. Also, if you look at the environmental versus clinical, you will find that quite a few of the war-related samples, they come from the environment, not only in the clinics. So we still have a lot of samples to go through. This is brilliant, but we needed to start answering questions immediately. So we went back to our previous idea, which was, how can heavy metals drive antimicrobial resistance whenever it comes to Acinetobacter baumannii and conflict? So we went back to the lab. We took Acinetobacter, a control strain. We started incubating it with heavy metals. We started with barium, with lead, with arsenic, with cadmium, and we started doing serial passages over several days, increasing the concentration of heavy metal bit by bit to try and match what's in the environment. Believe me, sometimes we never reached what's out there, but we did our best. And then at some point we would assess the antimicrobial susceptibility of the mutants that we are generating, and we see if something changes. And again, right, we were, something does change. If you look at this table, in your first column here, you have the profile of the control strain. That's Acinetobacter baumannii, non-mutated control strain that we have to buy and not isolate from any patient. Now, we started incubating it with lead, and bit by bit, we increased the concentration until we reached 8 millimolar lead. Mind you, in the environment after conflict, you're talking about concentrations that are 110, 150, and 200 millimolar. And to get to 8 millimolar, it took us 3 months. We are continuing to increase the concentration, but 8 millimolar is a number that gave us very good results, because look at cefepime became resistant, Piptazo, piperacillin tazobactam, gentamicin became resistant as well, and ceftazidime. The change is not very much, but we can take it with a grain of salt, but it became intermediate. So we took that sample, right? We cultured it again, and we went on to scanning electron microscopy, because we started seeing something strange on the agar. So when we did the scanning electron microscopy, over here you can see the standard Acinetobacter baumannii. How does it look under SEM? Once you have 5 millimolar lead, you can start seeing the bacterium changing, it's elongating. And then the 8 millimolar, it becomes more of a freak. So we weren't very sure. Did we do something wrong over here? So we extracted genomic DNA and we went on to sequence. And in our case, we're using both platforms, the long reads and the short reads, in order to get better view and close the genome completely. We identified mutations, and we took also more than one mutant per condition to average out the mutations. We identified mutations that deemed very interesting, and we are in the process of analyzing more. But before I talk about the mutations, we did the same thing with barium, and barium yielded even worse results than lead. And barium, by the way, is one of those heavy metals that you wouldn't expect to be in bullets and in shells and in rockets. But once you start reading about that stuff, hoping nobody will knock on your door at some point, you'd be surprised how much barium plays a big role in ammunition. When we got to 80 millimolar barium concentration, the Acinetobacter became resistant to carbapenem, to meropenem, which is one of the last resort antibiotics, to cefepime, to tazobactam, and to aminoglycosides. It became intermediate to one and resistant to the other. So we did the same thing. We did also scanning electron microscopy, and the change, or if you compare barium to lead, if you would just put those two images, you would think they are two different bugs, but it's the same. So we did also DNA extraction, and we started looking at the mutations from more of a functional side and a computational side. So where are we now, and what are we planning to do next? We are planning to complete the genome mapping very soon by comparing by actually extracting the mutations that we find in those mutants that we've generated, and trying to identify them into some of the clinical isolates. Mind you, in the last 48 hours, we've taken very few samples, and we did that exercise, and we can find them only in war-wounded patients. We cannot find them in non-war wounded patients or non-war wounded samples. The second objective we have is to go back out to the environment and resample again. Now, look for Acinetobacter baumannii, especially that one of our collaborators, Professor Muhammad Abyad, he already sampled post-war, and we know exactly how much heavy metals there is in the soil. So we're going to look for Acineto in the environment. And when we go back to the lab, we are also planning to do the same concentration of all the heavy metals in the soil under one condition and incubate Acineto with it and see what happens and what changes. So in conclusion, AMR and conflict helps you look at non-antibiotic drivers when it comes to AMR. And you focus as well on sentinel organisms like Acinetobacter baumannii and Vibrio cholera. I didn't talk about Vibrio over here, but Acinetobacter is the main character. This is helping in shaping the perfect storm. I didn't show you really what's happening in Ukraine, what's happening with the data coming out from from Syria with our collaborators that we are working with, but Acinetobacter takes over, especially in places like Ukraine and in Europe, where usually their percentage of Acinetobacter is a lot lower than ours on a happy day, whenever there is no conflict. And the jump they see is tenfold higher than the jump that we see. So the take-home message is, warfare creates evolutionary pressure, and we have demonstrated that. Co-selection does drive resistance in a way where we won't be able to control it at some point, especially that the mutations that we are looking at the moment are non-reversible efflux pumps, and it's not only something that can go away after time. Acinetobacter does exploit chaos, and whenever you have, so if you look, for example, at the data that came out from Turkey after the big massive earthquake, a lot of Acinetobacter clinical samples were isolated from people who were injured in that. Basically, there are several reasons not to cause war. One of them is not to drive AMR further to end on that note. And I want to thank my team who's been working on this really hard for the past few years. And thank you for your attention.
Thank you, Dr. Abu Fayyad. Next speaker is a virtual participation by Dr. Hiba Al-Haj.
Good morning, everyone. First of all, I would like
Dr.
to
Hiba
start by
Al-Haj
thanking
is
Drs.
an
Matar and
associate
Baibo for the
professor in experimental pathology, immunology, and microbiology. Her talk will be on reviving neglected battles.
Good morning, everyone. First of all, I would like to start by thanking Drs. Matar and Baibo for the kind invitation to present in the CIDR annual meeting. And our story today is about reviving neglected battles. Can inequalities change the story of parasitic diseases? We'll be talking today about Toxoplasma gondii, which belongs to the phylum Apicomplexa, regrouping a big number of parasites, including Plasmodium responsible for malaria. Starting with some generalities, Toxoplasma gondii is a highly prevalent parasite infecting 30% of the worldwide population. It has been classified in 2019 by the CDC among the neglected parasitic infections requiring public control. This parasite infects all warm-blooded animals, and in this case of infection, the spectrum of disease varies according to the immune status of the host. In immunocompetent individuals, the infection can be asymptomatic to mild symptomatic, while in immunocompromised individuals, it can be life-threatening if not treated. Worth noting that this parasite can cause congenital toxoplasmosis in primo-infected women, and the outcome of the infection varies according to the trimester in which the parasitic infection is encountered. If during the first trimester, this leads to abortion. If during the second or third trimester, we can see on babies a lot of symptoms, including hydrocephalus, intracranial calcifications, or chorioretinitis. The current treatment strategies against toxoplasmosis rely on general antiparasitic drugs, including spiramycin, pyrimethamine, sulfadiazine, among others. But these treatments cannot limit any damage if a baby has become infected, and all of them are unable to eliminate the brain cysts that are responsible for the chronic form of the infection that is happening in 30% of the worldwide population. As for the vaccine, there is no vaccine that is available for humans until today. The only vaccine called Toxovax is available to treat ovine, bovine, and cattle, and this is an attenuated strain that reverted to pathogenic strain, which limited the human use of this parasite. So until today, there is no effective drug or gold standard treatment for this disease, and there is an absence of an available vaccine for humans. If we take a look at the life cycle of this parasite, we find that TLRs, as in every pathogen, play a major role in the recognition of this parasite. And more importantly, TLR7 is one of the key TLRs that recognize the parasite in different hosts. The importance of TLR7 in the infection of Toxoplasma prompted us to test an agonist, which is imiquimod, and see whether it is effective against toxoplasmosis. Imiquimod is an FDA-approved drug that was used as immune response modifier to treat genital warts. In the context of parasitic infections, this drug is used as a second-line treatment with pentavalent ammonials against cutaneous leishmaniasis. Since the current treatment are effective against the acute form of the infection and none is effective against the chronic form which persists in hosts for a lifespan and which is the one which reactivates in immunocompromised patients, we were particularly interested in testing imiquimod against chronic toxoplasmosis. For that purpose, we allowed the chronic infection to establish in mice for 3 weeks, and then we treated for 1 month an optimized dose of 50 microgram of imiquimod every other day. After 4 weeks, we sacrificed the mice and harvested the brains. And when we harvested the brains, we were happy to see that imiquimod could significantly decrease the number of bradyzoids in the brain of infected mice. However, since we have some cysts that remain, the question was, are these cysts capable of reactivating? This prompted us to submit the mice to dexamethasone immunosuppression, and we found out that the ones that were infected with wild-type cysts were all dying from the reactivation, while the ones that were treated with imiquimod failed to reactivate. We, of course, dissected the full mechanism of action to see the mechanism that is dictating this efficacy of imiquimod against the bradyzoid cyst. And indeed, we have demonstrated that the cysts of bradyzoids that are persistent in neurons will reactivate back to tachyzoids when we treat with imiquimod. And this conversion is leading to the activation of innate immune cells, probably dendritic cells, leading to the upregulation of TLR 7, 11, and 12, which results in the MYT88 activation, followed by the MAP kinase activation, leading to the production of IL-12, then IL-1 beta, and interferon gamma, and this leads basically to clear the reactivated foci. We have then addressed an important question which is known in literature and that basically correlates toxoplasmosis with host-associated behavior, including a lot of disorders like depression, suicidal attempts, schizophrenia, among others. To tackle these questions, we have used rats that are excellent model to study behavioral changes. And we used a panel of behavioral tests. I'm showing only one today, which is the open field test. In this test, basically, when rats stay at the periphery of the box, it means that they are anxious. And when they move to the center, it means that they are exhibiting an exploratory behavior, which means that they have reduced anxiety. And we treated those rats with 3 types of drugs. After establishing the chronic infection for a month, we either treated with imiquimod or with sulfapyri, which is the golden standard clinically used combination of drugs, or with imiquimod followed by sulfapyri. So why did we test this combination? Because we proved that imiquimod is inducing reactivation. So we said that if it is reducing reactivation, we have tachyzoid stages, which means that if we apply sulfapyri, we are going to get rid of the forms that are causing the acute form of the infection or the reactivation of chronic toxoplasmosis. And the colors in here are shown here on these graphs. So we have tested the behavior of rats over 3 sessions. And over these sessions, I want you to look, please, over the 3rd session because it was the most significant one. If you look at the rats that are infected with toxo and that are untreated, you see that they are spending more time at the periphery and less time in the center, which means that they are exhibiting less exploratory behavior. If you go and treat with imiquimod, which is the red bar in here, you see that they spend less time in the periphery and they start moving towards the center. Now for imiquimod followed by sulfapyri, it was the best way, and this is where rats basically spend the least of the time at the periphery and the more of the time moving to the center to explore objects. And this is where we could prove that imiquimod-based therapy could reverse the anxiety-like behavior in chronic toxoplasmosis. Now, the problem of reactivation of toxoplasmosis following treatment with imiquimod was not very reassuring, although we could demonstrate that this was the way to dictate the molecular mechanism of imiquimod. In collaboration with medicinal chemists in Montpellier, we tested a panel of 49 imiquimod derivatives, and we showed that 3 out of which exhibited a strong activity against Toxoplasma gondii. And this is manifested in here by the reduction of tachyzoid stages. I will move directly to the chronic form of the infection because, again, this is one of our main interests in our lab. And we indeed found that this imiquimod that we called EAPB-08303 is really reducing the charge of cysts in the brain of infected mice more prominently than imiquimod. It reduces basically the transcript level expressed by bradyzoids, and more importantly, when we treat with this drug, we don't see any reactivation in the brain of mice. This is confirmed by real-time PCR and basically by Western blot, where we could not detect any tachyzoid marker. So this was for us very reassuring to find one of the imiquimod that is very effective against the chronic form of the infection and that does not lead to any cyst reactivation. And to dissect the molecular mechanisms of the drug against chronic toxoplasmosis, we have basically performed RNA-seq on brains of chronically infected mice, whether treated with imiquimod, whether treated with the EAPB-08303, or whether treated with the standard treatment that is sulfadiazine pyrimethamine. And to our surprise, we did not identify a huge number of genes that were changed. And if we compare, for instance, EAPB-08303 to imiquimod treatment, we found out that we have around 54 downregulated genes and only 15 upregulated genes. We looked more carefully into these upregulated versus downregulated genes, and we found out that the ones that are downregulated correspond mainly to pro-inflammatory pathways. And we, of course, chose some of these pathways and confirmed that the EAPB-08303 is suppressing this pro-inflammatory process. And if you can see in here, again, the EAPB-08303 is leading to the sharp and significant decrease in all the tested transcripts that are playing a key regulator process in the pro-inflammatory cytokine. Now, taking a look at the ones that were upregulated and specifically related to EAPB-08303, we found out one very important that stood out, which is ARC. What is ARC? ARC is basically an activity-regulated cytoskeleton-associated protein, and it is playing a major role in synaptic strength, and it is essential for memory consolidation and learning. And it is well documented that the suppression of ARC leads to the involvement of neuroinflammation, oxidative stress, and altered calcium signaling, which are all important in case of the parasitic infection, namely the chronic form of the infection. And to our attention, when we validated ARC expression following this RNA-seq data, we found out that EAPB-08303 is significantly upregulating ARC expression in the brains of chronically infected mice. So in conclusion, we did perform a qualitative or a comparative analysis between imiquimod and EAPB-08303. Basically, imiquimod has a moderate effect on cyst number as compared to 08303. The primary mode of action of imiquimod is the reactivation of bradyzoids, the activation of TLRs, the cytokine production, and this cytokine production is leading basically to the clearance of reactivation. The concern is, of course, the neuroinflammation in the context of immunocompromised patient. If we look at 08303, we see a more effective drug on the cyst number in mice with no reactivation. The primary mode of action is through suppression of inflammation, which promotes the cyst control, and more importantly, it is playing a major role in neural plasticity, which is a key regulator or a key pathway that is targeted by the infection upon establishment of the chronic toxoplasmosis. So this makes this drug safer if we move further steps towards the clinic and test it on immunocompromised patients. With that said, I would like to finish by thanking, of course, my team members who are working very hard to produce this nice data. And I would like to thank particularly Dr. Maggie Hamiye, who is a postdoctoral fellow in the lab and who did most of this work. I would like to, of course, thank my collaborators at AUB and most importantly at Montpellier, the medicinal chemist who did all these derivatives. And of course, Abdullah, who performed the amazing bioinformatic analysis, along with Dr. Muhammad Hamid from the GAR network, and of course, our intramural and extramural funding agencies. Thank you for your attention.
Thank you for Dr. Haj. Now, next speaker is Dr. Margaret Shrinian, who will be talking about epigenetic regulation in glial cells.
Thank you. Thank you, everyone. I would like to specifically thank those who stayed to the last session of this section, even though they didn't see a microbiology word or immunology in the title. So I'll show you today that I work on the epigenetics of brain development in the context of cancer. But all these regulations, the epigenetic modifiers that I'm going to talk about, they are fundamental in microbiology and immunology as well. And this also shows that I'm a real, a live example of diversity in our department and also in CIDR. So, one of the major problems in glioblastoma, which is a high-grade tumor, brain tumor, is the aggressivity of this tumor. And the aggressivity of this tumor, it doesn't come only from the uncontrolled proliferation of the cells, but it also comes due to the ability of these cells to invade healthy cells. So, and how does this happen? This happens when a tumor cell remodels itself so that it can degrade the extracellular matrix and invade the healthy areas in that region. So extracellular matrix remodeling is equivalent to glioblastoma aggressivity. And how can we understand this better? Okay, so one of the major components and the proteins or the genetic regulator that we are interested to look into is TET enzymes. So TETs are regulators of the chromatin. So what they do is that they demethylate the DNA so that it's more open for transcription. And in glioblastomas, we have seen that TET enzymes are mostly upregulated. And it's very rare cases where we can find mutations in TET enzymes. Most of the time, these are upregulations that we can see, and humans have 3 different TETs, TET1, TET2, and TET3. Sometimes TET come along with other mutations, such as histone mutations, TP53, BRAF, B600E, etc., and others. And this is different, this combination is different between pediatric GBMs and adult GBMs. So here is a small explanation to you on how TET functions. So TET, as I said, is a demethylase that demethylates the DNA. So it's a molecular eraser. During development, our chromatin is open, in an open state. That is to facilitate early neuronal and glial progenitor cells function, so enables to develop that brain plasticity that we need, and also to facilitate gene expression changes. So during this stage, the chromatin is very open. Then we have something called the transitional stage. So TET functions during this chromatin open stage because it removes the methyl groups, so the chromatin is more open. During the transitional state, what happens is that TET enzymes specifically bind special regions on the chromatin based on the need. Later on, when we have a closed chromatin state, where it should be, our chromatin should be mostly condensed so that transcription is not anymore possible, there will be a lot of DNA methyltransferases that will come into the picture to methylate our DNA, and these are molecular writers. DNMTs are molecular writers. So this is the whole process. And you want to have an open state so that you allow for cell proliferation and differentiation, and then you need to have that closed chromatin state during brain development to stabilize these neurons and glial cells. So we started looking into TET in our favorite model organism, which is Drosophila melanogaster. I'm not going to talk much about why did we use this model, but one of the main reasons is that Drosophila also has TET enzymes. It only has one TET, so it's easier for us to understand the function by just removing this gene and see what happens, but also due to the conserved function of epigenetics and epigenetic regulators that is between humans and flies. So previous postdoctoral fellow in our lab, Felice Frey, asked this question. First of all, she did some analysis and realized that Drosophila TET is very close to human TET3. And then she decided to remove TET from the flies by creating different genetic alleles and see what happens to the flies. So the complete removal of TET is very detrimental to the flies. So as you can see here, so the flies die early on. And this is the orange line, which is the TET null allele. You can see that the flies do not survive beyond larval and pupal stages. However, for our surprise, there were few flies that were hatching, and we call them escaper flies, that were hatching in these null mutants. And these flies die after 24 hours after they survive. And they are really few. But we were curious to see what are their phenotypes. And you could see here that these flies have difficulties in movement, so they move really not in a coordinated way. And when they flip on their backside, they're not able to straighten themselves. They always need the help to do so. And purposefully, we kept these flies ups and down so that they don't attach to the food because they're not able to move. And then we took them and visualized their movement. So this made a lot of sense. But then we said, okay, there is some coordination defects that probably is due to the brain and the motor function in the fly. Let's go back to early development and look at the larvae, which is one stage before the adult emergence, and see what happens to these flies. So a former PhD student in the lab, Joy Ismail, have taken the brains, and what she has done is that she knocked down TET. This is not a null allele anymore, so we're knocking down TET specifically in glial cells because we're interested in glioblastoma, which is a glial cell disease, so our focus will be glial cells. So she knocked down TET in glial cells, and specifically in something called midline glial cells. So this is the fly larval brain, and the green dots are the midline glial cells. And when she did that, she noticed that the axons that have to cross this line do not cross it properly anymore. So this is a wild-type fly brain, and this is the knockdown brain. You could see that there is a heart-like shape, so the axons, they're not going to where they should. Building on this, a current PhD student, Malaka Klet, she decided to see what happens if she knocked down TET in all glial cells, all differentiated glial cells in the brain. And when she did that, which is these green cells you could see, she noticed that there is an increase in the number of the glial cells. So, and this is really important for us because we want to see what's the role of TET. So we need to look into the mutants, we need to look into the knockdowns, but also we need to look into the overexpression of TET because it kind of resembles what happens in GBM. So what Malak has done is she overexpressed a full-length TET, Drosophila TET, into the brain of these animals. She overexpressed it in all differentiated glial cells. So this repo represents glial cell expression. So we drive DTET in all differentiated glial cells. And we could see this nice phenotype of the brain, which was 100% penetrant. So, and this is really a very unique thing to see. So every single brain that Malak has dissected had this elongated VNC, ventral nerve cord. This is a part of the brain that has to be decondensed during development. So what's happening is that this epigenetic regulator is inhibiting this condensation that should happen early during development. And you could see that the glial cells was lost in this overexpression experiments. To better understand what's going on, so we took these brains and performed transcriptomic analysis on them. So we did an RNA sequencing to look at the transcription level of different genes. We could identify a lot of upregulated and downregulated genes in the overexpressed brains. However, one of the interesting ones that we saw is MFAS, CON, which is CONTICI, and TIMP. And all of these genes are important for the remodeling of the extracellular matrix and for degrading the extracellular matrix in that brain so that the brain decondensation takes place. So this is not happening in our flies, and I think we could see that these genes that are important for that ECM remodeling were deregulated. Malak digged the literature a little bit because we were really fascinated by the phenotype, and we started to think what else we can find there. So what she could see, interestingly, that those genes that were upregulated in our data sets, such as TIMP, CONTICI, MMP, when they were expressed exactly in the same differentiated glial cells that she did, they had exact same phenotype. They had this VNC elongation. So what is it that is going on? So on your right side, you can see the VNC, and it's a schematic of the green cells where I'm talking about. These are the differentiated glial cells, which take a big part of the brain cells that are there. And they kind of provide that niche, that protection around which under it you have all the stem cells that are proliferating either into neuronal cells or glial cells. And it plays an important role. And this interactions between this glial niche and the neurons is very important. To show that this is this only for glial cells, or it can also, what is this phenotype coming it from? What is the origin of this phenotype? To do that, we overexpress TET early on in the stem cells that provide or then differentiate into glial cells and neurons. And when Malak did that, she could not see that elongated phenotype. So when she overexpressed TET in progenitor cells that give rise to the differentiated glial cells, none of these phenotypes were observant, meaning that the phenotype that we see is specific to differentiated glia. So when we looked into single-cell RNA-seq data, just to see where TET is expressed in a wild-type normal conditions, we could see that TET is almost in every cell in the fly brain. It's in the neurons, in glial cells, in midline glial cells, which are the circular cells that are wrapper-positive cells. And then when we look at the expression of these three important genes that I talked about that are part of the ECM, they are specifically found in the VNC and in these glial cells that are specific for that midline area in the VNC. So we have TIMP, CONTICI, and MFAS, as you can see here. To understand at the molecular level, how is this regulation of TET happening? How is TET regulating the ECM genes and ECM expression? We look into CHIP data, which is chromatin immunoprecipitation data, to see whether TET is binding TIMP. And as you could see here, that peak here shows that TET is indeed binding the promoter region of TIMP and basically regulating its expression. So then we went back to the patient data and we looked at samples, GBM patient data sets of 386 samples. And we compared the patient RNA-seq data with the fly glial cells in which we overexpressed TET. And we could see that there is many genes that were upregulated and downregulated in both, and there are some common genes that are in between. But one of the interesting things that we have found is that those genes that we saw in the fly glial cell to be upregulated, they also are upregulated in the glial cells of GBM patients, such as TIMP1, TIMP3, and FAS and CONTICI. So this is the hypothesis that we want to draw, but we're doing a lot more experiments to rescue that phenotype, to do some as well cut and tag analysis on the overexpression ones, because what I showed you is a ChIP-seq that is done on the wild type larvae, but we want to understand how TET is binding in when we downregulate it in the knockdown or the mutant and on the overexpression. But what we could tell up until today is that TET in the glial cell is regulating extracellular matrix genes, which are essential for proper glial niche presence. And once this is not happening properly, we have destabilization of the glial niche and deregulation of the extracellular matrix, which will increase basically the brain physics and the brain decondensation of the brain. Which will in turn lead to increased GBM invasiveness. And if we look at the literature, usually in GBM patients, the tumors, and as the neurosurgeons also explain, are usually much rigid than the surrounding parenchyma. And this has been tested in GBM cell lines as well. So this rigidity is somehow resembling what's happening there at the ECM level, and also it's preventing probably the immune surveillance on this, in this area where it should be recognized by the immune system. I will end up by talking to you a little bit about how we are using our Drosophila model to test for some specific compounds, to some epigenetic regulator inhibitors in the GBM context. So previously we have published in Molecular Cell an important piece of data on oncohistones and their role in regulating transposable elements and regulating the transcriptional regulation of genes that are important for eye development and brain development. So in there, we could see that a lot of P elements were upregulated, and this was really an important discovery because we showed for the first time that dimethylation of lysine 36M, which is an important chromatin activator, let's say, or a mark that is basically keeps the chromatin open and regulates transcription, affects transposable elements. And this is small data on the ChIP-seq analysis where we could see the comparison between the wild type and lysine 36 dimethylation, where we could see that there is complete irregulation of different classes of transposable elements on that chromosome arm. So we use that model, histone 3 lysine 27M oncogenes, because they are oncogenes in the fly, and we express them this time ubiquitously everywhere in the brain. Everywhere in the fly, actually, not only in the brain, in every cell. But if we do that, it's very detrimental, so the flies do not survive. To overcome that problem when we are doing our drug screening, we use a system called the GAL80 system. It's a very beautiful genetic system where we can regulate the expression of genes based on temperature. So at low temperature, we allow for the gene to be expressed, and when we shift the temperature up, so we allow for important developmental steps to carry on, and when we shift the temperature into higher 29 degrees, the expression will stop. So we used that fly line and we tested different, this is a pipeline that we developed in the lab. It takes a lot of time to do this, and there were a lot of students who were engaged in this. So our plan is to test for 150 epigenetic compounds that we think that in silico might rescue the lethality due to their interaction with the chromatin. So our readout is flies that are hatching, flies that are surviving this oncogenic expression. So these are the library creation of the compounds that we are going to use, and we have tested so far around 40 compounds, and there is one compound which showed a very promising outcome where the flies hatched, survived lethality, and we are right now screening the rest of the flies, the rest of the compounds, and validating this number 40 more in our experiments. So that's all, and I would like to end by thanking the people who have contributed to this work specifically. There are other projects in the lab where people are working. I did not mention all of them, and all the alumni of the lab. The work that I explained is mainly done by Malak Klit, which is a PhD candidate at AUB in my lab, and Sarah Mantaj is a postdoc. Jad Modi was a master student. He was working on the drugs. Abdel Majid Shahab was working on the drugs, and Bashir Tannous was working on the TAT brain project. Epigenetic drug screening, there are many students who also participated in this, and as well as faculty who helped me develop let's say, the strategic plan of how to move with this big number of compounds, Esper Saba and Antoine Abu Fayyad. And my collaborators at McGill University, Nada Jabado, in Sweden, Ruth Palmer. Together we're working as well on TET and ALK, Sanjay Kumar in Sweden, Angela Giagrande in France, and our Espers and Antoine. I also would like to thank Abdullah Al-Kirdi for all the bioinformatic analysis that he has helped us with, and all the funding agents who made this work possible. Thank you very much.
Thank you. Thank you, Dr. Shirinia. May the speakers please gather around the podium for questions. Any questions for the speakers?
Yes,
Yeah,
please.
Dr. Awo.
Yes, good morning, everybody. Thank you very much. First, I would like to congratulate the speakers and congratulate everybody on this great work, actually, under CIDR with Dr. Ghassan Beibu, and more so under yourself, Dr. Matar. Actually, we could see top-notch microscience research in here, which is really great. So congratulations, dear colleagues. It's really great. I have got two questions. First to Dr. Michel, please. Just learning from the experience with the RNA vaccines, which you have dissected and you showed us very clearly the time pattern of the immune response and protection. And so what can you conclude about RNA vaccines compared to other vaccines, let's say classical vaccines that are in use clinically? So what is the prospect for RNA vaccines that you can see, please?
Based on this limited experience with the RNA vaccine for SARS-CoV-2, it seems that it... Can you hear me?
Yes,
so based only on the SARS-CoV-2 vaccines, it seems to have a very strong, a much stronger immune response than the traditional adenoviruses and the protein peptide vaccines and inactivated vaccines. And then there are so many studies that started with the AstraZeneca vaccine, the adenovirus one, with one dose, very low response, and then they gave a second dose of mRNA vaccine from Pfizer or Moderna, and that boosted the response much, much higher. So I think mRNA vaccines, as far as vaccine is concerned, I think they have a very promising future. And I know a lot of companies, including Moderna, are working on so many different aspects of vaccines for pathogens, as well as vaccines for as anti-cancer treatment.
Thank you. If I'm allowed, Dr. Rahal, kindly, I would like to ask you about the EBV. Being one of the herpes viruses, but why is it not translated much into clinical practice compared to CMV, for example? We are very much concerned about CMV reactivation in our immunocompromised patients, but we do not generally pay much attention to the EBV. What would you say about that, please?
I think that's one of the difficulties with these viruses. So whenever CMV reactivates, it also reactivates other things with it at the same time. So it's always been kind of difficult to pinpoint whether it's this virus or that virus because they kind of reactivate at the same time, especially immunocompromised patients. So it becomes difficult to dissect clinically. And I think CMV gets the fame because it also mediates similar things. We've never looked at its DNA, and this is the opportunity we had with our studies to just take EBV by itself and examine it. I think that's one of the main reasons is that people have also talked about human herpes virus 6 as well as an important immunomodulator whose effects are under-tested and estimated, or understudied because it's kind of difficult. It also reactivates whenever CMV reactivates. So I'm not saying that it's only EBV that has these effects. And I think lots of these herpes viruses also have these effects, similar effects. And we've seen this with HHV6, where it also has these immunomodulatory effects. It doesn't mean that all viruses have these effects, but that family, those 8 viruses, they seem to have similar effects when it comes to immunomodulation and potentially stimulating certain aspects of the immune system that you do not want. Not to belabor the point, but also HHV6 and CMV are also associated with autoimmune diseases, and it might be along the same lines.
Thank you very much for this amazing presentation showing like top-notch research. I have 2 questions, if I'm allowed. One for Dr. Masad. So for your research on the immunology, did you look at spike-specific IgA, mucosal IgA in the nasal mucosa or in the saliva?
We didn't look at the IgA and the mucosa. There have been a couple of studies that showed very high levels of mucosal in the mouth, in the saliva, and even in breast milk. And they're mostly, the dimers are highly neutralizing, less so, they're more neutralizing than the IgG actually, but the monomers are less neutralizing. We didn't have access to those samples, but we thought that their levels in the blood will kind of give an indication of their potential level in the mucosal areas.
Thank you. My second question is for Dr. Antoine Abu Fayyad. So for your work in the war zones in the different countries in the Middle East, Libya and Yemen, did you find any correlation between the weapons that are used, like the bombs that are used, and the changes in the Acinetobacter, like between, for instance, what's happening in Syria, Yemen, Ukraine, and Lebanon?
Yes, we found, so that there are 3 things that we didn't really show. One, there is a clone of Acinetobacter that emerged from this region and has a signature, a genomic signature specifically for this region, and it emerged with conflict, and we could trace it back. It took us quite a while to actually draw it and demonstrate it and get it published, but we collected samples from 18 countries in the region, and we compared it to 90 countries outside the region to be able to show this. The second thing is there is a correlation between the type of weapon and the place the weapon is manufactured. So in order to understand the topic well, we had to do quite a lot of research in understanding how bombs and shells are made and bullets, where they are made, and if a country makes them, what are the prominent metals that are used? Because you hear it in a common language. We needed to understand at the chemical level why this is important, and we found vast differences and different metals as well. So that was the trick for us to actually crack the shell.
Any other questions?
Yeah.
Dr. Abu.
The new sonic wave weapons, they are bacteria friendly, as far as we know.
Dr. Abu Fayyad, I have a question, that the heavy metals that were done, in the lab, they induce the expression of the resistance genes. Now, is there a difference between those genes coming fresh from the environment and exposed to the heavy metals as compared to the hospital isolates? Is there a difference in the level of expression?
I'll be able to tell you on Monday. The analysis is running at the moment. So we actually did three things in parallel other than real-time PCR, because real-time PCR can be biased. We looked at the read counts and the sequencing while mapping the genomes, and it takes you a while because you have to annotate the genome first in all of them. And this is one of the things that we struggle with the IT system at AUB. You cannot really do big number of samples analysis at the same time. You have to either buy your own computer or split them into groups. So the analysis is taking a while, but will be done by Monday and we'll know next week.
Looking forward. Yes.
Congratulations to all speakers, really impressive work. A question for Dr. Bilane on the microbiota, and a lot of interest in this field, as you have shown. So in your analysis and the bacterium you've been able to identify, do you see any prospect for fecal transplant in ulcerative colitis patients with segregating with feces, high concentration in the bacteria that's lacking? Is this work being done somewhere? Because that would be a really good answer to the patient.
Yeah. This is actually, there are two papers out where they showed in animal models that performing fecal microbiota transplantation and ulcerative colitis modules will reverse and decrease inflammation. Now, the trick is that this is established, but the problem is with the FDA not approving the FMT, and they have the right to do so, because it's an uncontrolled environment. It has multi-drug resistant bacteria that can not be detected by sequencing due to the depth bias. And also, they cannot control the toxins inside the stool, nor the metabolites in the stool. So FMT will support UC patient, yes. It has been shown to be able to do so, yes, but it's not a safe approach. What we are trying to do here is creating the microbial community, we can't say the essence of the stool, but the actual thing that works in the stool, but in a controlled, safe environment to give it to the patient.
Thank you. If I may, another question to Dr. Abu Fayyad. So Tony, what we've noticed over the years is the more in the clinical, I'm going to translate what you find in the lab to the clinical setting. The more the Acinetobacter is becoming virulent, the less mortality we're getting with these patients. They just become colonized forever. We can't get rid of this colonization, but in terms of the clinical impact, mortality, complications, it seems that over the years we've seen a decline. And the question is, do you think that increased production of these virulent factors upon exposure to various things, including metals and others, are making the bacteria less virulent?
So we see that in Acineto, but we see the opposite in Klebsiella. And correct me if I'm wrong, Klebsiella patients are, quote unquote, apologies for the term, dying a lot more than Acineto patients lately. So this attributes to two things, and I know one of the fellows is presenting on Klebsiella later on. So if a bacterium makes mutations to become resistant to antibiotics, this has a price. We call that fitness cost, right? Because it mutated, it's going to become slow to multiply. So at some point, bacteria will start doing other mutations elsewhere to compensate this cost that it's paying. And there has been publications that showed the other mutations that the bacteria is doing has affected its virulence, not because directly from AMR, but through an indirect circle of trying to compensate the heavy AMR burden. Virulence is changing. You cannot say in a good direction. Its virulence is changing. So we don't know. It's sporadic. It can go in either way.
Okay, no more questions. Thank you. We have a coffee break up there in the hall, and we can resume at 12:25.
علي.
هذا علي. Okay, so we'd like to welcome you for the second session of the CIDR annual meeting. To start the session off, we're going to start with Dr. Ali Taubi, who will talk about oral step-down therapy for gram-negative bacteremia, the INVEST trial, international multi-center study. Dr. Ali.
Thank you. So good morning, everyone. I want to thank you all for coming on a Saturday. And I want to take a few moments to thank my mentor, Professor Soha Kanj, for giving me this opportunity to stand in front of you guys. So now we're shifting the gears. We'll be talking about clinical medicine. And today I'm going to be presenting the INVEST trial, which is the early oral step-down therapy versus continuing the IV therapy from uncomplicated gram-negative bacteremia. So we're going to start now. So as we know, gram-negative bacteremia is rising globally and remains a major cause of morbidity and mortality. So the most organisms isolated from blood cultures are particularly E. coli and Klebsiella pneumonia. So the majority of patients with gram-negative bacteria initially receives IV antibiotic therapy, and it remains unclear whether we can step down safely to oral antibiotics. And although the efficacy is still limited, oral antibiotics have several advantages. So the advantages of oral antibiotics, they eliminate the risk of catheter-associated adverse events. They enhance the patient quality of life. They also eliminate IV-related risks such as phlebitis, thrombosis. It also enables mobility, early mobility, and reduce also the length of stay in hospital. And it also reduces healthcare costs and resource utilization. This current management of gram-negative bacteremia entails a duration of IV therapy with limited evidence to guide oral conversion. The study aims to evaluate the clinical efficacy and economic impact of early step-down to oral antibiotics. So prior studies suggest that the oral therapy may be safe and cost-effective. Here we have the A-class trial, which shows similar clinical cure rates between oral and IV arms with shorter hospital stay and lower medical costs in the oral groups. Here we have another study which was led by Tama in 2019, which also showed after involving nearly 5,000 cases, they also demonstrated comparable 30 days mortality between oral step-down versus continued IV therapy with significantly shorter length of stay in the hospital and the oral group. So given the limited randomized prospective evidence across broader sources of gram-negative bacteremia, the INVEST trial was designed. So the objective of the study, the INVEST trial, is that the clinical efficacy is to evaluate the clinical efficacy and the economic impact of early step-down to oral antibiotics versus continuing IV therapy for clinically stable or critically ill patients with uncomplicated gram-negative bacteremia. For this, the first objective is to check whether the oral antibiotic therapy will be non-inferior to continuing IV antibiotic therapy and the primary outcome of 30-day all-cause mortality, and if it will show significantly lower health resource and service utilization costs compared with continuing IV therapy. So for this, we're going to be talking about the trial design. So First of all, this study is a multi-center, international randomized investigator-initiated, not pharmaceutical, where eligible participants must be clinically stable, non-critically ill in patients over the age of 18, which is adults, and of course with gram-negative bacteremia. So now let's consider two groups. The group that has been randomized into standard arms and intervention arms. So in the intervention arm, we're gonna, after randomization, a one-to-one randomization, whether they stay on oral therapy or IV antibiotic therapy. So the first group, if the patient falls into the intervention arm, they will start directly, immediately, the oral therapy using either fluoroquinolones or trimethoprim sulfamethoxazole based on their susceptibility. So, and the other group, when they fall, for example, into the IV therapy, they will stay at least 24 hours in the hospital, waiting for the doctor's decision to make whether they continue on IV therapy after 24 hours or they switch into the oral antibiotic therapy. So now, here are the participating sites. We said that it's a multi-center international, so here's some of the sites. And it's great to note that the AUBMC is the singular only center in the Middle East. And it's, of course, led by Professor Sohakanj. Here are the others in Malaysia, occupied Palestine, Italy, Spain, and Greece. So now we'll be talking about inclusion criteria and exclusion criteria. In order for the patient to be included in this trial, so they have to be an adult above 18 years of age. We have to get at least one sample of blood culture for positive for gram-negative bacteria. Also to be able to be randomized within 72 hours index of blood culture collection, and the latest pit bacteremia score, which has to be less than 4. Here's the pit bacteremia score on the right. And also we give the patient a written informed consent, which comes in 2 different languages that we give in AUBMC. It comes in English and in Arabic. So now the exclusion criteria are broad and there's a lot of them, but mainly we exclude the patient using these, and like we compare it to a score. So, for example, if the patient had undrained abdominal abscess, we put 1 point. Central nervous system abscesses, 1 point. For example, if he has sepsis, it leads directly to the complete exclusion of the patient. If he is also severely immunocompromised, also we exclude him directly. Also, if the patient can't absorb oral medication. So now for the plan of assessment and collection of outcomes. So the eligibility assessment, we start by it. It's including any pregnancy test if applicable. A written informed consent, which we talk about, which comes in 2 different languages, documentation of demographics, documentation of charts and comorbidity index and antibiotic history since admission, a physical examination, a radiographic findings if any are available, of course, a complete blood count and white blood cells, AST, ALP, CRP, etc. And the blood culture results. And this, the last one, the AQ5DL, we call it here. It's a quality of survey. We usually, and we always call the patients on day 14, day 30, day 90 to check up on them if they are clinically stable, if they don't have any adverse events of the drug, if they can continue their daily exercises, study, work at home, etc.
Ali, you have 2 more minutes.
Yeah. So we study also the primary outcome and the secondary outcome. The primary outcome is to all-cause mortality at 30 post-randomization patients for the standard arm versus the intervention arm. For secondary outcomes, all-cause mortality at day 14 and day 90 to study the adverse events, for example, C. difficile, catheter-related infections, change in the treatment strategy, or readmission. For statistical analysis, primary analysis will be performed on modified intention to treat, where all the patients are included into the study whenever they are eligible to the criteria and started the medicine. So if upper limit of 95% confidence interval falls below the 6% non-inferior margin, non-inferiority will be confirmed and concluded for intervention arm. So now for the secondary outcomes. The secondary outcomes usually we check the length of stay in the hospital, whether it lowers or extends. The mean difference and its 95% confidence interval will be provided for secondary outcome measures that are on interval scale, and comparison between study arms will be done using a two-sample t-test. Now here we can see on this graph that the study began on May 2022 and it's still until today. So the main goal is to reach the target of 720 recruitments in all the centers. So we wish to finish all these things at the end of April 2026 in the worst case scenario. So here, to go back to Lebanon, here at the IUB-MC, until now, we recruited 29 patients, where 18 of them are enrolled into the study, which makes the total enrollment rate of 62%, which is amazing. And we're still in it, and we're still recruiting patients until today, and hoping to continue this until the end of the study. Now, last but not least, a finding of non-inferiority in efficacy of oral fluoroquinolone and TMP-SMX versus IP standard of care antibiotics may hypothetically translate to wider adoption of a more cost-effective treatment strategy with better quality of life outcomes. Thank you for listening.
Thank you, Dr. Ali. The next speaker will be Dr. Ghaida Abu Daher. She will be talking about the epidemiology, characteristics, and treatment outcomes of carbapenem-resistant Enterobacterales bacteremia, a 4-year retrospective study.
Hello, good morning, everyone. Thank you, everyone, for being here, and thank you, Dr. Kans, for this opportunity. In today's presentation, I'll be talking about epidemiology, characteristics, and treatment outcomes of a carbapenem-resistant Enterobacterales bacteremia. This is a 4-year retrospective study done at IUB-MC. So as an introduction, carbapenem-resistant Enterobacterales, which will be referring as CRE during this presentation, represent a significant group of gram-negative bacteria in the Enterobacterales order. Notably, we have E. coli, Enterobacter species, and Klebsiella pneumonia. These organisms have developed resistance to at least one of the carbapenem's antibiotics. They were previously non-existing before the 1990s, after which CRE have emerged as a major global threat over the past decade, significantly reducing available treatment options. As a result, WHO classified these pathogens as critical priority pathogens in 2017. So among CRE, Klebsiella pneumonia was the first organism to spread on a global scale, becoming endemic in the US, Greece, and Italy. A bibliometric analysis of 1,671 publications over 10 years identified Klebsiella as the most commonly isolated organism worldwide. Carbapenem resistance in CRE is primarily due to carbapenemase production. There are 5 classes of carbapenemase categorized into 3 classes, as we know, class A, B, and D. CRE infections are mainly associated with healthcare facilities and range from catheter-associated infections to VAP. Despite the significant burden of the CRE infection, optimal treatment remains largely undefined, with evidence primarily derived from case report, case series, and small retrospective studies. Empiric treatment of CRE in cases of gram-negative bacteremia mainly depends on the patient's history, our local resistant pattern, and our epidemiology. There is no evidence-based data to guide empirical coverage of these MDR organisms. Options may include high-cost drugs or high-toxicity drugs, or a combination with carbapenem and colistin, tigecycline in combination with another agent, or ceftazidime avibactam with or without aztreonam. Once CRE is identified identified, the antibiotic regimen is adjusted based on the organism susceptibility profile, definitely, although data on the efficacy of combination therapy versus monotherapy and specific antibiotics conferring higher survival rates are limited. So as for the methods, this is a single-center observational retrospective study. We used AUBMC EPIC system and electronic health records to review the records of patients who were infected with CRE bacteremia between the 1st of January 2018 and the 1st of January 2024. The aim of the study was to detect the incidence and the trend of CRE bacteremia at AUBMC per 1,000 patients per day between the end of 2018 and early 2024, explore the antimicrobial susceptibility of CRE and its evolution, identify the mortality rate in patients with CRE bacteremia at AUBMC, determine the effect of medical therapy on mortality in such patients by comparing outcomes in patients receiving monotherapy versus combination therapy, and compare mortality of CRE bacteremia and hematologic malignancies versus other subgroup of patients. So as for results, from 1,129 patients with Enterobacteriaceae bacteremia, we had 73 CRE bacteremias in 53 patients. Time to bacteremia was 17.6 almost days since admission. 81% of patients used carbapenems within the 3 months before having a CRE bacteremia. Peripheral blood cultures were not taken in 4%, negative in 8%, and positive in 88% of patients. The mean time to positivity was 12 hours. Central line was present in 29 out of 73% of patients, which is a 38.4%, and time to positivity from the central line was 10 hours. So our highest number of CRE bacteremia was during 2022 with 14 bacteremias due to E. coli, followed by 10 bacteremias in 2023 for E. coli and 5 for Klebsiella. Most of our bacteremia originated from the GI tract, from pneumonia, and from UTIs. Carbapenem, CRE bacteremia and Enterobacteriaceae isolated were resistant to carbapenems. Ceftazidime avibactam MIC was sent for 58 bacteremias and showed susceptibility in only 13.8%. To note that this study was done before the introduction of Carba5 at our lab, which we concluded after that our CREs are NDM positive and not susceptible to zavicefta-astrionam. 77.8% of bacteremia were diagnosed while the patient presented to ED or regular floor, and 22% of bacteremia were diagnosed in patients already in closed units or for more than 48 hours. Length of hospital stay where bacteremia occurred was 37 days. Time from admission to bacteremia was 17 days. So 56% of patients were alive at day 30, 36.5% were dead at day 30, and 6% lost follow-up. Many patients had previous ICU hospitalization, at least one hospitalization prior to the CRE bacteremia. So these two tables are comparative for mortality in terms of empiric single versus empiric double therapy. So the 30-day mortality was very similar between the two groups, approximately 40% in both empiric and double therapy. Also for hematologic malignancy, the mortality was 19 patients were dead out of 55, which is 35%. And 50% for the patients who are non-hematology. So non-hematologic patients had a higher 30-day mortality compared to hematology patients. This is the resistance profile for CRE. Of course, 88% were resistant to all classes of carbapenems, 14% were resistant to colistin, 74% were resistant to amikacin, 17% were resistant to zavicefta and zerbaxa, and 56% were resistant to tigecycline. Patients were mostly of hematologic malignancy and of solid malignancy. So for the discussion, the highest rate of CRE was observed in 2022, which decreased in the following 2 years, mainly by application of firm infection control measures and in terms of screening and isolation. Most of patients used carbapenems within 3 months, which is a non-risk factors, among others, for development of CRE bacteremia, as described in a study done on hematology patient in 2017 that showed that risk factors included many options as recurrent ICU admissions, prior antimicrobial exposure, prior CRE isolate, or BSI during neutropenia. So I'm going to go back a little bit to molecular. I used these slides from Dr. Antoine Abou Fayyad's analysis on whole genome sequencing. So in these tables, we can see the plasmid and the genes responsible for resistance. The majority of Klebsiella were in sequence type 3A3, which is a hypervirulent organism. And these isolates were from our hospital exclusively, and they were all Klebsiella. 91% have expression of both OXA48 and NDM5 together at the same time. This table shows the antimicrobial susceptibility testing done among the 18 mentioned Klebsiella isolates, and it's comparable of what we found also in our lab results. And these are the AMR genes that are found in Klebsiella. There are many of them. There are similar ones between the 18 isolates. Some of them are show resemblance, others just don't. And that's it. Thank you.
Thank you. Excellent. Now, the next talk is by Dr. Ayam Al-Husari, who will be speaking about the immunity gaps and seroprevalence of measles, a retrospective single center study from Lebanon.
Good morning. So I will be talking about the seroprevalence of measles, which is a retrospective single center study at a tertiary care center in Lebanon. Measles remains to be a threat to the Lebanese population, with outbreaks still occurring to this date and vaccine coverage remaining suboptimal. The study provides an evaluation of the seroprevalence of measles in the Lebanese population, while also focusing on possible contributing factors. It will aid in filling major gaps in knowledge about the measles immunity status in Lebanon and will improve understanding of recurrent outbreaks. So the primary outcome of the study the primary objective is to determine the seroprevalence of measles in a sample population tested at AUBMC between 2011 and 2022. Secondary objectives include determining the fraction of seronegative subjects by age groups and to explore possible reasons for seronegativity, to determine the geographic distribution of seronegative subjects and to correlate with known rates of vaccine uptake, to examine the effect of the COVID-19 pandemic on the measles seropositivity, and to identify the range of measles IgG levels in women with childbearing potential and in infants under 9 months to inform vaccination policy in Lebanon. So this was a retrospective study done at AUBMC, so monocentric, where IgG and IgM serology results from January 1, 2011 to December 31, 2022 were included. The data was collected from hospital medical records, and the ECRF form included age, gender, geographic location, presence of chronic diseases, immune status, and vaccination history. Inclusion criteria were all males and females who had a measles IgG and IgM test ordered at AUBMC. Exclusion criteria were those who had unavailable medical records for review. So the study was approved by the IRB at AUBMC. Patients' names or other information were not used, and the data was collected from the charts on electronic systems such as EPIC and the HIS portal. CRF was completed electronically through REDCap and transcribed into a secure Excel sheet. So as for the results, 1,399 individuals were included, 53% were males and almost 47% were females. Mean age was 27 years. 95% were immunocompetent and only 5% were immunocompromised. 52% were from Beirut area and 30% from Mount Lebanon. The rest were distributed across North and South Lebanon, Kisrwen, Jbel, Bekaa, and Baalbek, or not residing in Lebanon. So overall, 60.4% of individuals had positive measles IgG test results. For the stratification by age group, the highest seropositivity rates was in the age group of 41 to 60 years, and the lowest was in those less than 11 months old. However, there was very limited data available for this age group, where we had only one individual younger than 6 months, and the IgG level was 0.53, so equivocal. As for women of childbearing age, 31.5% were seronegative. The IgG levels ranged between 0.22 and 5.93, and the average was 1.64. The outbreaks years were noted in 2013 and 2014. The highest IgG seropositivity was in 2022. So after the COVID-19 pandemic, we noted a spike in the seropositivity rates. Vaccine compliance, we had only 161 subjects with available vaccination records. Out of those, 78% were compliant and around 22% had delay in vaccination. Overall, we have a suboptimal immunity where 40% of individuals in the Lebanese population remain susceptible to infection. Highest seropositivity being in the elderly notes that we still have a shortage of the more than 95% immunity that is required for herd protection. And the WHO data shows that there is low vaccination coverage in Lebanon, so our results are in concordance with that. For infants who are younger than 11 months, early waning of maternal antibodies reported in the literature may let us think about considering moving the zero-dose vaccine to 6 months rather than 9 months. And as for women of childbearing age, we can conclude that there's a need for screening and booster vaccines. We noted a high rate of seronegativity in Beirut and Mount Lebanon. However, this could be attributed to the population density. And the lowest rates were in North Akkar and Baalbek Hermel governorates, which is according to WHO. A rise in IgG was seen in COVID-19 after the COVID-19 pandemic, so this may reflect catch-up campaigns or improved awareness. So in conclusion, these findings reveal significant gaps in immunity in Lebanon across age groups. These data underscore the need to strengthen vaccination efforts, consider targeted catch-up strategies for high-risk populations to prevent future outbreaks, and to move the zero dose of the vaccine to 6 months instead of 9 months. Thank you.
Thank you, Dr. Hayam. Next talk is by Dr. Dima Khes. She will talk about BCG scar reactivation in Kawasaki disease, results from the Pan-Arab Kawarabi survey and a scoping review of the literature.
Good afternoon, everyone. I would like first to introduce the team working on this project. So this project is a multidisciplinary collaboration between pediatric cardiology and pediatric infectious diseases. It was led by Dr. Najib Dahdah, Dr. Shwaiter, and Dr. Harahshe, who are pediatric cardiologists and international leaders of Kawasaki disease, in collaboration with the CIDR at AUB, represented by Dr. Wakim. So as you know, Kawasaki disease is an acute systemic vasculitis of childhood with predilection for the coronary arteries. If left untreated, unfortunately, 25% of children experience coronary aneurysms without IVIG. However, early recognition and early treatment can lower this risk to less than 5%. Kawasaki disease is a diagnostic challenge for every physician because we don't have any specific diagnostic test. Diagnosis is mainly clinical. It's established by the presence of fever for 5 days with the following associated mucocutaneous features in classic forms. However, the challenge is even more pronounced in infants, where incomplete forms can present, which increases the risk of delayed recognition and delayed treatment. So mortality can reach up to 0.2%, especially from cardiac death due to missed or delayed diagnosis. As we know, Kawasaki disease is mainly a disease of the Asian ancestry, and most data derive from East Asian populations. This is why an initiative, Kawarabi, was established to generate evidence and improve Kawasaki disease outcomes in the Arab world. So Kawarabi stands for the Kawasaki Disease Arab Initiative. It's a multi-center collaborative that was established in 2021 to improve Kawasaki disease outcomes in the Middle East and North African region. It connects experts in Kawasaki disease across 13 Arab countries and it aims to establish patient registries on Kawasaki disease, conduct education and more awareness about this diagnosis, and advocate for resources like IVIG availability. So Lebanon participates in Kawarabi through 2 academic centers, including AUBMC. Now, as infectious disease specialists, we were particularly interested in exploring the BCG site reactivation in Kawasaki disease. So as you know, the BCG or Bacille Calmette-Guerin vaccine is typically used for tuberculosis prevention in TB endemic areas, which applies to most of the Arab countries, except for Lebanon, which is a non-endemic area for TB. However, we often provide care for patients who are immigrants or who receive the BCG vaccine abroad. And typically, it's administered in the first year of life. So since 1997, many reports have described the presence of BCG site inflammation or BCGitis at the vaccination scar during the acute phase of Kawasaki disease. And this has been hypothesized as a possible early diagnostic sign for Kawasaki disease, specifically in infants who usually present with incomplete presentations. So as we can see, the BCG site reactivations in Kawasaki disease can present with redness, crusting, and induration at the vaccination site. And it has been actually incorporated into the Japanese 2020 diagnostic guidelines for Kawasaki disease, but it remains under-recognized in other parts of the world. And it's especially prevalent in young infants less than 1 to 2 years of age. So the data on the BCG scar reactivation in Kawasaki disease is limited, especially in BCG vaccinated populations beyond the East Asia. This is why 2 complementary projects were conducted under the scope of Kawarabi. The first one is a pan-Arab BCGitis survey, where we try to assess the real-world recognition of BCG scar reactivation in Kawasaki disease. So we distributed a survey across 13 Arab countries in order to map the BCG vaccination policies, which differ between one country to another, and to identify clinical practices and the diagnostic value of this particular sign. And the second part was to conduct a scoping literature review on this sign to synthesize the global data on BCG reactivation in Kawasaki disease in order to determine the prevalence, the clinical timing, and the diagnostic significance of this sign. So first, the survey was a cross-sectional survey-based study. It targeted physicians who usually diagnose and manage Kawasaki disease. So we had a high response rate reaching around 98%, and a total of 356 physicians with a wide geographic distribution who care usually for patients with Kawasaki disease across 13 Arab countries. Most of the respondents were general pediatricians, but 20% were pediatric cardiologists, 7% rheumatologists, and infectious disease specialists. So most of the physicians reported that the BCG vaccine was mandatory in their countries in 95% of the cases, and it was administered before 1 month of age in 88% of cases. So the striking finding was when we asked the physicians if they verify the BCG site while caring for patients with Kawasaki disease, only 50% of physicians were aware about the sign, whereas the others do not regularly check the BCG vaccine scar. And among those who do check the BCG site, 24% have encountered BCGitis or BCG site inflammation in Kawasaki disease cases, and the usual age groups and 64% was infants less than 1 year of age. So there is a spectrum for BCG site activation features, but the most common was BCG site pain, tenderness, and itching, and less commonly, and erythema, and less commonly purulent discharge or abscesses. We also tried to explore the relationship between BCG site reactivation and different indicators of disease severity. So only 6% of respondents observed that BCGitis usually occurs in the absence of skin rash, and 4% only reported resistance to IVIG among patients who had BCGitis, with also a low rate of coronary artery aneurysm prevalence and 3% of shock or severe disease in patients with BCGitis. And typically, 54% reported that BCGitis usually resolves after 1 to 2 weeks. In the second part, we conducted a scoping literature review to look for the global literature on BCG scar reactivation following the PRISMA guidelines. So we searched 2 large databases And among 177 records identified, we ultimately included 30 case reports and series and 10 institutional retrospective cohort series about BCGitis. However, we could not conduct a systematic review or meta-analysis because, as you see, the literature was dominated by case reports and series, and there was a marked heterogeneity in the definitions and outcomes for Kawasaki and for BCGitis. So the publications span around 25 years. The first report was in 1997, and then we can see every year we have 1 to 2 publications about this topic. And we tried to map the countries per WHO region. And as expected, 17 publications were from the East Asian region, less so in the Americas and in Europe, and 3 publications from the Middle East region, 2 from KSA and 1 from Turkey. Now, among the 10 institutional retrospective cohort series, 33.1% was the mean prevalence of BCG scar reactivation in Kawasaki disease patients. And among those, the coronary artery aneurysm rates were often reported as lower in the group who actually displayed BCGitis. However, this finding did not have any statistical significance, and we need more prospective cohorts with appropriate controls in order to establish this relation.
Dima, 2 minutes.
Yeah. So in case reports and series, we identified 36 well-documented cases of BCGitis in Kawasaki disease. So most of the children were less than 12 months of age with a mean of 8 months. Most of them had incomplete Kawasaki disease. And importantly, children usually had fever around 7 days before receiving IVIG, while the BCG reactivation site appeared around day 4 to 5 from fever. So it really can be used as an early diagnostic tool for Kawasaki disease. Now, one reported case of mortality was reported in Japan in 2015, where a 5-month-old has died because he actually presented with only fever and rash and BCGitis, and it was not recognized as Kawasaki disease until post-mortem autopsy. So it can usually present most commonly as erythema and induration, and less commonly as crusting, scab formation, ulcerations or erosions, vesicles, pustules, and more rarely suppuration and lymphadenitis. There are 3 main theories behind the BCG site activation in Kawasaki disease. The actual pathogenesis is still unclear. The first theory is immune recall or delayed type hypersensitivity at the site of the residual mycobacterial antigen. The second theory is molecular mimicry or cross-reactivity between mycobacterial and human heat shock protein, the HSP65. And finally, and the most plausible theory, is cytokine-driven amplification, because as we know, Kawasaki disease is characterized by elevated cytokines, which can trigger cutaneous and vascular inflammation at the BCG site. So to conclude, we can say that the BCGitis has a high diagnostic yield and clinical utility of BCG site reactivation in Kawasaki disease. It usually appears in infants who are most likely to exhibit this sign, probably because it's closer to the immunization age. And incomplete Kawasaki cases predominate in infants who are most needy for supportive diagnostic signs. Usually, the reports show lower or non-higher incidence of coronary artery aneurysm and IVIG resistance in this group. However, we believe that there is a need to increase physician awareness about this sign and possibly integrate the BCG site reactivation into regional and international diagnostic guidelines, especially when caring for BCG vaccinated children. And finally, I can proudly say that we have presented this project in Dubai back in November, and we will hopefully present it in the International Kawasaki Disease Research meeting in Amsterdam in March 2026. Thank you.
Thank you, Dima. The next talk is by Mr. Saab Shihab, our medical student who is currently outside the country. He'll be talking virtually about nasopharyngeal carriage rates, prevalent serotypes, and antimicrobial resistance of Streptococcus pneumoniae in Lebanon, a national cross-sectional study.
Good afternoon, everyone, and thank you to the Center for Infectious Disease Research for the opportunity to present at this year's meeting. My name is Saab Shahab, and today I'll be presenting our work on nasopharyngeal carriage, serotype distribution, and antimicrobial resistance of Streptococcus pneumoniae in Lebanon. Despite medical advancements, Streptococcus pneumoniae remains a leading cause of pneumonia, meningitis, bacteremia, sinusitis, and otitis media, particularly among young children. In fact, carried serotypes are associated with both invasive and non-invasive disease, and disease often follows acquisition within carriers themselves or from asymptomatic carriers. As such, carriers are reservoirs for transmission, and measuring carriage actually informs prevention. Through this study, studying nasopharyngeal carriage, it helps us understand disease epidemiology as well as inform prevention strategies, including vaccine policy and antimicrobial stewardship. In the Middle East, several regional carriage studies exist. However, Lebanon-specific data remains limited. A 2023 systematic review examining pneumococcal carriage and resistance patterns across Western Asia highlighted a notable lack of updated data from Lebanon. In the absence of contemporary national surveillance, it becomes very difficult to tailor vaccine strategies, anticipate serotype replacement, and monitor emerging antimicrobial resistance trends. Through this study, we aim to fill this important knowledge gap. Taking this into account, we designed this study with 4 key objectives. First, to estimate pneumococcal carriage rates and compare them across age groups under 5 years. Second, to describe circulating serotypes stratified by age, region, vaccination status, and daycare attendance. Third, to characterize antimicrobial resistance patterns to commonly used antibiotics. And finally, to assess vaccine-related patterns, specifically comparing vaccine type versus non-vaccine type serotypes. This study's methodology was a national cross-sectional study conducted among children under 5 years of age residing in Lebanon. Children who were actively receiving antibiotics at the time of sampling were excluded. Recruitment also occurred across multiple settings, including daycares, nurseries, and schools, while children under 3 were primarily recruited from clinics and outpatient departments, taking into account that some children may not be attending daycare and nursery. In addition to nasopharyngeal sampling, we contacted parents after sampling and completed a structured survey capturing demographics, vaccination history, and relevant clinical and environmental factors. Before discussing the statistics, I wanted to briefly highlight the fieldwork that made this national dataset possible. And with the support and coordination of Ms. Nati Horani in CIDR, together with infectious disease fellows and medical students, we were able to conduct fieldwork across multiple sites throughout Lebanon, reaching 472 children across the nation. This cohort of 472 children was predominantly older, with the majority aged 16 months and above, while the remaining participants were distributed across early infancy. And this distribution allowed us to examine carriage and serotype patterns across key developmental and exposure windows. When it comes to geographic distribution, participants were recruited across multiple Lebanese governorates, with the largest contributions from Mount Lebanon at 30.7%, followed by Nabateeh and North Lebanon at around 20%, ensuring broad geographic representation. It's important to note that the number of schools and recruitment sites approached in each region was informed by population distribution data in Lebanon, allowing sampling effort to broadly reflect regional population density rather than convenience alone. With regards to vaccination status and dose timing were documented for nearly all participants, with most children vaccinated prior to sampling. This distinction of vaccine timing was characterized relative to sampling to avoid exposure misclassification and ensure that only doses capable of influencing carriage were included in future analyses. Across all doses pre-sampling, PCV was the predominant vaccine formulation, with more limited uptake of PCV20 and minimal use of PCV10 and PCV15. Importantly, this pattern was consistent across sequential doses, indicating relative homogeneity in vaccine exposure within the cohort rather than dose-specific shifts in vaccine type. Interestingly, in contrast to the pre-sampling profile, PCV20 and PCV15 make up a larger share of vaccines administered after sampling. However, as previously discussed, these post-sampling doses are not directly relevant to carriage exposure, as they occur outside the biologically meaningful window for that analysis. What would be more informative then, from an epidemiological perspective, is identifying which children were up to date at the time of sampling, based on age-appropriate pneumococcal vaccine schedules. both of which differ based on the Ministry of Health, which follows a 4, 6, and 12 to 15 months, versus the private schedule that can range from 2, 4, 6, and 15 months. And noting that some children may be vaccinated in private settings while adhering to the Ministry of Health schedule, that can further complicate the analysis. In short, for the time being, while post-sampling doses were therefore excluded from exposure analysis, I presented them here purely to provide context on evolving vaccine availability and prescribing practices, and to highlight possible emerging shifts in vaccine uptake within the population. With regards to the baseline clinical characteristics of the children, the cohort was predominantly healthy, with low prevalence of chronic comorbidities, minimal regular medication use, and infrequent recurrent hospitalizations. This slide also summarizes key feeding and environmental exposures relevant to pneumococcal carriage. The majority of children had a history of breastfeeding, providing a baseline of early life immune exposure, and in contrast, a household smoking exposure was common, reflecting a prevalent environmental risk factor within the cohort. Both variables are very well-established modifiers of respiratory colonization and were captured to allow adjusted analysis of carriage and resistance patterns. Finally, with regards to the demographics of these children, overall, higher educational attainment was common among both parents, with university education being the most frequent category, particularly among mothers. Moving on to our laboratory workflow, it is currently ongoing with the support of Mr. Fatah Al and under the supervision of Dr. Fatima Daroub. It begins with molecular detection using PCR targeting the SP2020 gene, and only PCR positive samples will proceed to culture. To date, this has yielded 22 culture plates positive for Streptococcus, and from each positive plate, up to 3 distinct isolates are selected for downstream analysis, although in some cases, unfortunately, fewer isolates were recoverable. This particular approach allows us to assess potential multiple serotype carriage within a single sampling. Afterwards, serotyping is performed primarily through Illumina sequencing, with multiplex PCR reserved for SP2020 positive samples that do not grow on culture. Antimicrobial resistant profiles will then be derived directly from sequencing data, enabling high-resolution resistance characterization. To summarize, we have collected data and samples from 472 children, enrolling children under 5 across multiple Lebanese governorates. Detailed pneumococcal vaccine exposure and dose timing have been systematically captured for most participants, and importantly, the dataset is now finalized and ready for linkage to carriage, serotype, and antimicrobial resistance patterns, positioning this study to generate contemporary, locally relevant evidence for pneumococcal surveillance and vaccine policy in Lebanon. Lastly, I would like to acknowledge and thank our study team at AUB and the Center for Infectious Disease Research, particularly Dr. Hassan Tbaibo, Dr. Noor Youssef, Ms. Nancy Hourani, Dr. Selina Boutros, Dr. Fatima Daroub, and Mr. Fatah Al, alongside all of the infectious disease research fellows and students who made this study possible. We sincerely thank the participating families and children, as well as all collaborating recruitment sites. With regards to funding, this work was supported by the AUB Faculty of Medicine through my scholarly concentration track, as well as additional support from the Center for Infectious Disease Research Fund of Dr. Hassan Tbaibo. Thank you for listening and wishing everyone a great day.
Thank you, Saab. So the next talk is by Dr. Fatima Daroub. She will be speaking about genomic characterization of invasive disease causing Streptococcus pneumonia in Lebanon, data from 2003 to 2025.
Sure. Okay.
Yeah. Why?
Sorry.
Hi everyone, today I will be presenting our results on the genomic surveillance of Streptococcus pneumoniae in Lebanon, a study that has spanned 2 decades of collected samples. So Streptococcus pneumoniae is a troublesome pathogen. It is able to cause invasive disease and it is associated with high burden. For example, back in 2015, it was estimated that around 3.7 million episodes of IPD have occurred in children. And the good news is that we have vaccines, pneumococcal conjugate vaccines, that have actually been enrolled and that could decrease this burden. And as you can see in the figure, the mortality associated with IPD has also decreased as more kids got vaccinated. Now, as of August 2024, PCVs have been enrolled to around 166 countries, including Lebanon. In Lebanon, we have PCV7 implemented in 2006 to the private healthcare sector, PCV13 in 2010 also to the private healthcare sector, and then PCV13 was incorporated into the expanded program on immunization in 2016, which made it available for children from the public healthcare sector. So, as mentioned, we conducted the first genomic analysis of invasive Streptococcus pneumoniae in Lebanon, and these samples were sequenced and collected as a part of FLIPSIP, which is an ongoing program that conducts surveillance of IPD. Usually, we enroll patients who were hospitalized with invasive disease. Both sexes are included and all age groups are included. Specimens in this analysis, which I will be showing today, were collected from 35 different hospitals across Lebanon, covering all regions. So we were able to sequence 277 isolates using either long or short read sequencing. We analyzed our data using the GPS pipeline, and only 4 samples failed analysis QC. So we ended up including 273 genomes in our analysis. So we divided the study into 3 periods. This is 3 periods. This is according to when the vaccines were implemented. For example, EPI PCV13 represents the period after the incorporation of PCV13 into the expanded program on immunization. And it is the latest study period. So we found that overall serotype 3, serotype 14, and serotype 19F were the most prevalent serotypes. And in the latest study period, so EPI PCV13, around half of the genomes were non-vaccine types. Also, in the latest study periods, the most prevalent lineages were GPSC12, GPSC10, and GPSC6. Among GPSC12, GPSC12 only consisted of serotype 3, and similar to what has been found in global reports, GPSC12 expressing serotype 3 did actually expand after vaccine implementation. Moreover, we found that after vaccine implementation in the latest period, GPSC10 consisted mostly of serogroup 24, which is known to be very highly resistant and virulent. We also found that due to vaccine implementation, we have now diversification of GPSC6. In the previous study periods, it only consisted of vaccine types, but now it also contains non-vaccine types such as serogroup 11. We also analyzed the distribution of vaccine types and non-vaccine types over the study period, and we found that PCV7 serotypes have decreased with time and across all clinical manifestations. We also detected a significant increase in non-vaccine types over the study period, which is what's really expected. This is similar to what has been observed globally. And when you actually introduce vaccines into a population, this does not only affect humans, you're also affecting the pneumococcal population, and you are exerting selective pressure on these isolates. We also analyzed IPD-associated mortality. We found that mortality was associated with the older adults group. We also found that PCV7 serotypes were associated with higher mortality rates compared to non-vaccine types. The recent replacement of PCV13 with PCV10, specifically pneumocele, in the expanded program on immunization might actually decrease the vaccine coverage of circulating serotypes. As you can see, during the API PCV13 period, which is the latest study period, the serotype coverage for PCV10 was only around 27%, which is around half of that of PCV13. Ideally, we should use, among licensed vaccines, we should use PCV20 because it has provided the highest coverage in this analysis across all clinical manifestations. So we conducted phylogenetic analysis and we examined the AMR trends in our population. We found that non-susceptibility to penicillin was highest, followed by cotrimoxazole. We found that certain lineages actually drove MDR in IPD patients in Lebanon. For example, GPSC1, GPSC9, GPSC61, and GPSC14 consisted only of MDR isolates. We also found that the majority of GPSC10 isolates were also MDR. We found that the majority of non-MDR genomes were also PCV13 or NVTs. And as you saw in the previous graphs, NVTs increased with time. So this might explain why we have a decreasing trend in AMR across 7 different antibiotics and over the study period. You can also note that the rate of multi-drug resistance has decreased with time.
2 minutes, Fatima.
Yes. So this study improved our understanding of the genetic structure of invasive Streptococcus pneumoniae in Lebanon following PCV introduction. Some of its strong points is its multi-center design and the extended period of surveillance. Some of its limitations is the limited sample size when we wanted to do subgroup analysis of individual GPSCs or serotypes. And not all available samples were sequenced due to resource constraints. Also, it would have been much better to get, for example, vaccination data from the patients and try to correlate that with circulating serotypes. So as a conclusion, this study highlights the importance of whole genome sequencing because without it, we wouldn't have determined the lineages, pneumococcal lineages in our population. And in fact, this is the first study from the MENA region to do so. And given the high mortality rate in the older adults population, it would be also beneficial to include them in future vaccination programs. And most importantly, in the latest study period, PCV10, which is now being used in API, does not provide sufficient coverage for circulating serotypes in Lebanon, and we should consider switching to PCV20. Finally, I would like to thank all of our collaborators and all of our funders and the CIDR team and the Sanger Institute team who contributed to this study. Thank you all for listening and attending.
Thank you, Fatima. The next talk will be by Dr. Sarwat Farhat, who will talk about the burden of invasive group A Streptococcus infections, a retrospective single tertiary care center experience in Lebanon.
Good afternoon, everyone. So strep pyogene is a common community-acquired pathogen that causes a variety of infection ranging from asymptomatic carriage to life-threatening diseases. And data from the MENA region is limited concerning the burden of group A strep diseases. A safe, effective, and affordable vaccine to prevent infection and their most serious complication is needed. In 2019, two initiatives marked a positive step toward gas vaccine development, the Strep A Vaccine Global Consortium and the Australian Strep A Vaccine Initiative. data to help formulate the decision on vaccine recommendation, the best age target, when and how often, the economic feasibility are lacking. So the primary objective of this study was to describe the burden of invasive group A strep disease at AUBMC. The secondary objective were to describe patient characteristics, to identify risk factors, to determine the site of infection and clinical presentation, to determine the hospital length of stay, the need for ICU admission, mechanical ventilation, surgical intervention, if there's any superimposed complication, and time to mortality or hospital discharge. This was a retrospective monocentric chart review study dating from the 1st of January 2019 till 31 of December 2023 included, and it was approved by the Institutional Review Board at AUBMC. So 4,913 patients admitted diagnosed possibly related to invasive group A strep. Patients with non-invasive group A strep infection were excluded, and only 47 patients met the inclusion criteria. In the next slide, I'll be showing the preliminary result for 40 cases, and 7 new retrieved cases will be later on added to the analysis. So among the 40 patients diagnosed with invasive group A strep infection between 2019 and 2023, the majority of patients were adults and the mean age was around 54 years of age, and only 12% were pediatric patients with a mean age of 4.5 years of age. We retrieved a lot of comorbidities associated with invasive group A strep infection, including but not limited to cardiovascular diseases and diabetes. And for the predisposing factors leading to invasive group A strep infection, we can see that there's skin conditions, recent surgeries, and steroid intake that increases the risk of invasive group A strep infection. And when looking at the distribution of invasive group A strep throughout the years, we can see an outbreak of the invasive group A strep post-COVID-19 pandemic, and mainly in the second quartile of 2022, mainly between April and June. And cases were clustered predominantly during late summer and fall month, with the highest monthly count was observed in August. And when looking at the diagnosis associated with invasive group A strep, we can see that soft tissue infection with bacteremia and sepsis were the most common diagnosis associated with a 37 and 27% respectively, and the case fatality rate was around 15%. And for the frequency of surgical intervention, 100% of patients with necrotizing fasciitis required surgery, compared to 25% of patients with septic arthritis and 66% of patients with osteomyelitis. For the sampling site, 100% of the patient had a positive culture. One case was reported to have a positive rapid strep infection from a wound culture, and another one was positive for 16S RNA genome sequencing from blood sampling. And when looking at the antimicrobial susceptibility, we can see that penicillin was 100% sensitive, and the clindamycin resistant rate was 27%. And after doing univariable analysis to see the correlation with ICU admission and mortality, we can see that cardiovascular diseases and diabetes mellitus was significantly associated with increased ICU admission, but cardiovascular disease was not associated with, or not statistically significant association with death, compared to diabetes mellitus and renal or liver diseases that were significantly associated with an increased mortality rates. So moving on to the discussion, outbreaks of invasive group A strep infection in Lebanon beginning mid-2022 following a period of low incidence during COVID-19 pandemic restrictions. These findings align with the literature post-COVID-19 invasive group A strep resurgence in the late 2022-2023. Seasonal trends related revealed that invasive group A strep cases clustered predominantly during late summer and fall month, unlike the other studies showing the prevalence of invasive group A strep during winter and early spring. Whereas for the clindamycin resistance in our study, the rate was 27.5%, which was similar to the CDC surveillance report from 2022 that showed that 1/3 of invasive group A strep isolate were resistant to clindamycin. Outbreak of invasive group A strep infection in Lebanon beginning mid-2022 coincided with the global outbreak following a period of low incidence during COVID-19 pandemic restrictions. And it affected both children and adults with a predominance of older adults with chronic comorbidities. Skin and soft tissue infection with bacteremia and sepsis were the most common clinical presentation, with a case fatality rate 15%. Comorbid conditions were significantly associated with increased ICU admission and death. And thus the urgent need to strengthen the early recognition and management protocol to reduce severe outcome in high-risk population, as well as the importance of preventive strategies, including vaccines. Thank you.
Thank you, Sarwat. The next talk will be by Dr. Soleil Farhat, molecular identification assay for the rapid diagnosis of invasive fungal infections in hospitalized patients.
17.
Good afternoon, everyone. Thank you, Dr. Baibu, for your continuous support, and I hope you're all enjoying the conference. I'm very honored to be presenting today one of the most important studies we are conducting at the center. It's focused on the early identification by molecular assay of invasive fungal infection. First, I would like to disclose that this project received support from Pfizer. As we all know, invasive fungal infections, or IFIs, are a major global cause of morbidity and mortality. Yet they are often under-recognized. They mainly affect hospitalized patients, especially those who are immunocompromised, and can cause high fatality rates where outcomes strongly depend on early diagnosis and early initiation of antifungal therapy. Globally, over 1 billion people are affected by fungal disease, including about 150 million with severe infections, and they can cause an estimated of 1.7 million deaths each year. Importantly, this burden continues to rise with the expanding population of immunocompromised patients and the limited therapeutic options available. Despite the burden associated with IFIs, their diagnosis still remains a clinical challenge. The diagnosis currently still relies primarily on culture and histopathological examination, which are considered the traditional reference standards. However, these methods have several important limitations. Culture-based diagnostics are often associated with long turnaround times. Often they take several weeks for definitive results. The method can also exhibit low sensitivity, particularly in patients who have low fungal burden or who are already on antifungal therapy. Histopathological diagnosis, on the other hand, usually require deep tissue biopsies, which is not always clinically feasible. As a consequence, IFIs are often diagnosed later in the disease and sometimes post-mortem. As we've seen, conventional diagnoses have a relatively low clinical yield, and therefore there is an urgent need for rapid, sensitive, and specific diagnostic tools to enable early targeted antifungal therapy. Our study had two main objectives. The first was to develop a rapid and efficient molecular assay that could detect IFIs early, ideally before patients experience severe clinical deterioration. The second objective was to assess the diagnostic performance of this assay, specifically its sensitivity and specificity using the in-house molecular test we developed in our laboratory. This study was designed as a prospective cohort study. The study population included patients at risk or with confirmed IFIs. This encompassed patients from both genders and from all age groups and were willing to provide a written consent. On the other hand, people who are unwilling to participate were consisted the exclusion criteria. The primary outcome was the diagnostic accuracy of our in-house molecular assay, specifically its sensitivity and specificity in detecting IFIs compared to conventional methods.
So why the molecular approach? Our rationale was based on molecular approaches which offer a strong solution for detecting IFIs because they allow the direct detection of genetic fungal material and can enable earlier identification even when fungal burden is low or if the patient has already been receiving antifungal therapy. One of the most widely used molecular targets in fungal diagnostic is the ribosomal RNA gene cluster. This region is highly conserved across fungal species and exists in multiple copies per genome, which enables the sensitivity of the test. As shown here in the figure, no pointer, okay. We can see that this RNA cluster is consist of 18S, ITS, and the 28S region. The 28S RNA region provides an optimal balance for use. It contains conserved primer binding sites while also including variable domains that help us with the species and genus level identification. For this reason, we specifically targeted the 28S rRNA region using the well-established NL primers that can amplify a broad range of clinically relevant fungi. As for the technique, the first step of our molecular approach is to extract the genetic material of the fungi from the clinical specimen used. Then fungal DNA undergoes PCR amplification targeting the 28S rRNA gene using the NL primers we previously talked about. The PCR products are sequenced using Sanger sequencing, which provides high quality and accurate data for definitive fungal identification. Whenever available, the molecular results are also compared with conventional culture findings to assess concordance and to highlight the need for the rapid detection, especially when cultures are delayed. And finally, we evaluated the assay performance by calculating diagnostic sensitivity and specificity. Phases 1 and 2 focused on validating the 28S PCR assay, where in phase 1 we performed analytical validation using the pure fungal isolates. We tested a total of 61 samples with 59 spiked with different fungal species and 2 unspiked for negative controls, with the lab team being blinded to the sample content. All spiked samples were correctly identified and the negative controls remained negative, demonstrating the excellent accuracy. Phase 2 extended this validation to remnants of clinical samples. In culture-positive samples, the assay detected fungal isolates in 30 out of 36 cases, corresponding to 80% detection rate. In culture-negative samples, the fungal DNA was still detected in 6 out of 76 samples, including organisms such as Malcesia restrica, Candida kaffir, and Trichosporin species. Phase 3 represented the clinical evaluation of the 28S in the real-world patient samples. Between January 2024 and November 2025, we screened a total of 567 patients for eligibility. Out of these, 251 were excluded mainly due to incomplete documentation or insufficient volume for the sequencing. This resulted in 316 patients enrolled for 28S PCR testing. Completed information were available for 311 patients. Positive PCR specimens of the 28S were available in 62 patients, accounting for 19.9% positivity, and negative specimens were a total of 249 with a total of approximately 80.1%. 28S can detect a wide range of clinical samples with percentages shown above. In our samples, we used different types of tissues, including deep airway samples, sterile fluids, including abdominal fluids, pleural fluids, CSF, blood samples, all types of clinical samples mainly. For the results, the overall mean age of the cohort was around 53 years. Using regression analysis, and when looking at the age groups, we found a significant association between age and IFI positivity with P equal to 0.029. Patients aged between 65 and 80 years had a significant higher proportion for positive 28S PCR compared to younger age groups, and they almost showed a threefold higher odds of IFI detection with p-value equal to 0.002 in contrast to pediatric population and younger adults, which is clinically relevant as older patients usually have more comorbidities. As for the gender distribution, males constituted the majority of the study population and demonstrated a significantly higher rate of IFI positivity compared to females. Patient accounted for 79% of positive specimens, while females accounted for 21%, with this reaching statistical significance of P equal to 0.002. This also was supported by existing literature, which states that males are more prone to IFIs, mainly due to the difference of immune regulation by difference of hormonal sex hormones present. I won't go over all of these individual numbers. I just wanted to point out that in our cohort, all the participants had at least one comorbidity with a significance in renal impairment and liver disease. We also examined risk factors for IFIs. Overall, most traditional immunocompromising factors were also present. We then examined the underlying immunocompromising conditions among PCR-positive patients. Many were receiving immunosuppressive therapy or had either hematological or solid malignancies, which highlights the vulnerability of this group for IFIs. For the diagnosis performance of the 28S PCR, and when considering all clinical samples, the assay showed a sensitivity of 56% and a high specificity of 91.5%. To note that the overall sensitivity is reduced by samples with very low fungal growth of approximately 100 organisms per milli, which the assay could not detect. This low growth samples may represent contamination or colonization, which should be clinically correlated. When blood samples were excluded from the analysis, sensitivity increased to 62%, while specificity remained high at 91.45%, suggesting the non-blood specimens are more suitable for fungal detection using this assay. The 28S PCR assay, I just wanted to highlight here its ability to detect a wide range of fungal species. We see among the most prevalent ones Candida albicans, Candida glabrata, Candida tropicalis, and other Candida species like Candida auris, among others. It was also able to detect Aspergillus, Marcisia species, dermatophytes, and other mixed results. In this slide, I just wanted to note that in some PCR-positive wild culture negative organisms, we were able to detect Candida albicans, Candida tropicalis, Marcisia restrica, and other, which shows the higher sensitivity of the PCR towards these organisms. To conclude, the 28S fungal molecular identification assay shows a strong identification as a rapid and broad range tool for detecting IFIs. It complements conventional culture by identifying uncommon and culture-negative organisms, addressing key diagnostic limitations, and these prelim findings that we just talked about today support its role in enabling earlier and more accurate diagnosis. Our ongoing analysis will further evaluate the turnaround times and clinical impact, particularly in relation to the timely initiation, optimization, and possible de-escalation of antifungal therapy. Following the successful completion of both phases 1 and 2, and now we are currently concluding the phase 3 of the study, we are still enrolling patients until this assay will be clinically implemented, hopefully very soon. Thank you so much. And special thanks to Dr. Fatima Dakroub and Dr. George Arash for their continuous support on this project as well. Thank you.
Thank you, Soleil. Next talk is by Dr. Mona Qasim. She will be talking about the associations of RSV viral load with symptoms and clinical outcomes in patients with severe acute respiratory infection.
Good afternoon, everyone. So today we'll be talking about the associations of RSV viral load with symptoms and clinical outcomes. So as we all know, respiratory syncytial virus, or RSV, is a major cause of severe acute respiratory infections. And severity of RSV infection can range from a mild illness to a severe life-threatening lower respiratory tract involvement. And highest risk groups include infants below the age of 6 months, older adults, and patients with chronic comorbidities such as asthma and COPD. So each year, RSV causes a major global burden, with millions of hospitalizations reaching almost 3.6 million hospitalizations for those who are aged below 5 years, and substantial mortality in both infants and adults. Despite this substantial global burden of RSV-associated SARI, predictors of disease severity at presentation remain poorly defined. Disease severity is known to be influenced by host factors such as age and comorbidities. However, the contribution of viral factors is less clearly understood. So, RSV viral load reflects the extent of viral replication in the respiratory tract and has been proposed as a potential marker of disease severity. So, we conducted this study with the aim to characterize RSV viral load kinetics in relation to symptom onset, to examine associations between RSV viral load and patient characteristics and clinical presentation, and in order to evaluate the prognostic value of RSV viral load for disease severity and adverse outcomes. Moving on to the methodology. So this study was conducted as a part of the Global Influenza Hospital Surveillance Network, or GIHSN, which is a multinational surveillance platform, which includes sites from both hemispheres, where Lebanon is the only participating site from the Middle East. So GIHSN is a network which involves more than 100 hospitals in more than 25 countries worldwide. It's co-funded by the Foundation of Influenza Epidemiology, and it's a multi-center prospective hospital-based active surveillance program, which aim to collect epidemiological data and virologic data on influenza and respiratory viruses. So in Lebanon, this study was conducted across 6 participating hospitals, representing both urban and rural settings and covering broad population catchment areas. And currently, GIHSN is expanding its network to involve a total of 7 hospitals. So we conducted active year-round hospital-based surveillance where daily admission lists were screened across all age groups to identify patients within the previous 24 to 72 hours who met a predefined set of conditions, including acute respiratory infections or other influenza-related presentations. And eligible patients were recruited using the extended SARI case definition, which includes having an acute respiratory infection with cough occurring within 10 days requiring hospitalization. Clinical and demographic data were collected and samples were tested using real-time PCR for influenza, RSV, and COVID. So a total of 526 patients were involved. Between October 2021 and May 2025, a viral load was quantified using real-time PCR with a standardized RNA calibration curve, and RSV viral load stratification cutoff was 7.24. As for the results, as for the demographic and clinical characteristics, almost 60% of our population were males. Most of them, almost 80%, were aged below the age of 5 years, and 21% had chronic conditions. So the most 2 common reported symptoms were nasal congestion and malaise. Mean hospital stay was 6 days. 7% of the patients developed complications. 22% required ICU admission, and our mortality rate was 1.5%. So to characterize viral kinetics in RSV infection, we have evaluated viral load in relation to time since symptom onset. So as we can see here, we observed a modest but significant negative correlation, indicating that viral load generally decreases over time. And most of our patients were tested within 5 days of presentation. Also, mean viral load declined significantly over the first 5 days. However, we observed a higher mean at day 6, likely reflecting a subset of patients with delayed viral peaks. So we next examined the association of the mean RSV viral load with some key demographic and clinical characteristics, where viral load did not differ significantly by gender or the presence of chronic conditions. However, we observed significant differences by age, especially those who were aged above 65 years old. Also, higher viral loads were present in patients with diabetes and COPD. Also, we can see that viral load did not differ significantly by the presence of renal impairment, neoplasms, or the presence of viral or bacterial co-infection. However, patients with rheumatological diseases had significantly higher viral loads. In contrast to patients who received antibiotics, during admission, they had significantly lower viral loads. We also examined the relationship between RSV viral load and symptom presentation. So patients who were tested within 3 days of symptom onset had significantly higher viral loads in contrast to those who were tested later on, which is consistent with early viral replication. Importantly, higher viral load was associated with lower respiratory tract symptoms, mainly dyspnea and wheezing. When we examined other systemic symptoms, we observed a different pattern. Patients presenting with malaise, confusion, or lethargy had significantly lower viral loads. This suggests that these manifestations may be more reflective of host response rather than direct viral replication. Finally, we examined clinical outcomes among patients hospitalized with RSV-associated SARI according to the RSV viral load, and we can see that patients with higher viral load had higher rates of ICU admissions, complications, oxygen supplementation, pneumonia, and bronchiolitis, and they had higher need to be admitted in high dependence units. As for the predictors of RSV-associated complications, we can see that in univariate analysis, several factors were associated with clinical complications, including older age, high RSV viral load, and some comorbidities. However, in multivariate analysis, high RSV viral load was the only factor independently associated with the occurrence of complications. So in conclusion, higher RSV viral load was associated with increased clinical severity and complications, but not with prolonged hospitalization. These findings support RSV viral load as a clinically relevant virological correlate of disease severity, and RSV viral load may serve as a potential prognostic marker. However, it still requires further validation in prospective studies. And at the end, I would like to thank all CIDR team members, the previous and current members, and special thanks to Dr. Fatima Dakroub and Dr. Hassan Zaraid. Thank you.
Thank you, Mona. We have 2 more presentations to go. The next one is by Dr. Samar Delli. She will be talking about genomic and clinical epidemiology of SARS-CoV-2 in Lebanon, a prospective multi-center study between 2020 and 2024.
Good afternoon, everyone. So I'll be presenting a recently published article. Recently, by 3 days, it was published in the BMC Infectious Disease Journal, entitled Genomic and Clinical Epidemiology of SARS-CoV-2 in Lebanon, a prospective multi-center study 2020-2024. So, as you all know, SARS-CoV-2 emerged in 2019 and caused significant global morbidity and mortality. Early in the pandemic, older age and comorbidities were identified as key factors for severe disease and death, shaping public health responses and vaccination priorities. Over time, the clinical trajectory of COVID-19 has evolved due to viral evolution, widespread vaccination, and immunity from prior infections. Distinct SARS-CoV-2 variants have been associated with differing clinical severity. For example, Delta was linked to more severe disease, while Omicron was associated with milder outcomes and fewer hospitalizations. In addition, genomic surveillance is essential to understand variant-specific clinical impact. In Lebanon, long-term data on clinical and viral genomic epidemiology remains limited. Local assessment is needed due to differences in population immunity, vaccination coverage, and variant circulation. So this study was done with the aim to evaluate COVID-19 epidemiological clinical characteristics in Lebanon over 4 consecutive years, to identify mortality risk factors, explore age-related differences, compare SARS-CoV-2 positive and negative SARI cases, and contextualize clinical outcomes within the evolving genomic landscapes of SARS-CoV-2. So this was a prospective multi-center observational study. It was conducted in 6 hospitals across Lebanon from November 2020 to October 2024. The samples were collected as part of the GIHSN, which was presented by my colleague Mona Qasim. So included were patients who were hospitalized for 1 night or more and enrolled within 72 hours of admission. As for case definitions, so pre-season 2021-22, the ECDC modified ILL definition was used, and from season 2021-22 onward, WHO extended SARI definition was used. Excluded were the ones who were unable to consent or were hospitalized within 30 days. Data was collected through case report forms, and patients were followed until discharge or death. So for lab testing, respiratory samples were collected, multiplex RT-PCR was done, SARS-CoV-2 positivity was defined as a CT value less than 38, and whole genome sequencing was done on samples who had a CT value less than 28. And for the statistical analysis, so descriptive and comparative analysis was done, Kaplan-Meier was done for survival analysis, and multivariable logistic regression was used to identify mortality predictors. So from November 2020 to October 2024, enrolled was a total of 7,708 patients. 1,302, around 17%, were COVID-19 positive. And the surveillance year was defined as starting from November and ending in the following October. The highest SARS-CoV-2 positivity was during the season 2020-2021. So this slide and the following one shows the SARS-CoV positivity rates throughout each season. So in season 2020-21, there was 2 distinct epidemic waves. The first one was seen in Jan 2021 with a positivity rate of 64%, and it coincided with the health system strain and critical bed shortage nationwide. The second one was in Feb, with the launch of the first national COVID-19 vaccination campaign by the Ministry of Public Health. The second season, 2021-2022, showed also 2 distinct epidemic waves. The first one in Jan, with a positivity rate of 31%, coinciding with the initiation of a second nationwide COVID-19 vaccination campaign that was led by the US and AUB. And in August 2022, with a positivity rate of around 33%. In the following 2 seasons, as you can see, there was a transition to a sustained lower level of SARS-CoV-2 circulation without clear epidemic peaks. So when compared to other viruses, for example, influenza, COVID-19 did not demonstrate a clear seasonal pattern. Instead, there was recurrent peaks, especially observed in July and in August, likely driven by the influx of expatriate Lebanese returning from different regions during the summer. This table shows the demographic and clinical characteristics of the patients enrolled throughout each season. So across the 4 study years, there was a significant shift in the demographic and clinical profile of hospitalized COVID-19 patients, documenting the transition of the COVID-19 pandemic towards a flu-like epidemic starting from season 2022-23. The mean age decreased markedly with a parallel rise in pediatric cases, while vaccination coverage, particularly 3 doses or more of COVID-19 vaccine, increased significantly. Clinically, all markers of disease severity, including ICU admission, oxygen requirement, mechanical ventilation, pneumonia, ARDS, mortality, declined sharply over time. These findings reflect the combined impact of vaccination, viral evolution, and changing in the population immunity. So the most severe clinical outcomes were seen in the first season, which was 2020-21, And through the phylogenetic analysis in Lebanon, and from worldwide, it showed that the Delta variant was predominant in that season. And later on, this variant was replaced by the Omicron variant, leading to a progressive decline in disease severity starting from second season 2021-2022. This table compares the patients that were enrolled with COVID-19 and those who were negative. So the patients with COVID-19 were significantly older and had a higher burden of comorbidities, including cardiovascular disease, diabetes, and malignancy. COVID-19 was associated with significantly worse outcomes, including higher ICU admission, oxygen and vasopressor requirement, mechanical ventilation, sepsis, ARDS, pneumonia, and mortality. This slide shows the COVID-19 outcome by age group. So the green column represents the patients who were 18 years and younger, the pink, the ones who were 18 to 59, and the blue who were 60 years and above. So as you can see, the patients who were 60 and above had significantly higher ICU admission, pneumonia, mechanical ventilation, and mortality. While the pediatric patients that are represented in green, they had lower mortality and higher viral co-infection rates. This table shows the independent predictors of COVID-19 mortality determined by the multivariate analysis. So patients who were 18 to 59 and those who were 60 years and above had a significantly higher odds of mortality compared to pediatric patients. And the ones who received 3 or more COVID-19 vaccine doses also had a lower odds of mortality, and the ones with confirmed chest X-ray showing pneumonia had significantly increased in odds of mortality. This Kaplan survival plot shows the occurrence of pneumonia and the need of mechanical ventilation. And as you can see, both of them are associated with higher mortality in patients who had COVID. Lastly, this figure shows time-resolved phylogenetic tree displaying SARS-CoV-2 genomes collected from SARI patients in Lebanon, which were 138 specimens, and the sequences from global circulation, which totaled 567. The three nodes are colored by country. Lebanon is represented by the pink circles, while worldwide by the gray. So through this analysis, we saw that several distinct clusters of Lebanese genomes, including those in clades 22B and 22F, suggesting that more than one introduction may have led to local community transmission chains. And lastly, I want to thank all the team for your amazing work. Thank you.
Thank you, Samar. And our last talk for the day, Dr. Amani Haddara. She will be talking about SARS-CoV-2 in hospitalized children, epidemiology, characteristics, and outcomes 2020 to 2025.
Good afternoon, everyone. So I'm glad today to share our data about COVID-19 in children. So building on what my colleague Samar have said, we will talk about COVID-19 in children, and we'll talk about a population that was initially thought to be less affected, which is children. So since the emergence of COVID-19, children and adolescents have generally experienced milder disease compared with adults. However, pediatric infection has been widely documented worldwide, and its epidemiology have evolved progressively with multiple variant waves. Notably, the proportion of pediatric cases increased significantly from early pandemic from 5% to 10 to 23% during the Omicron period, with more than 16 million cases confirmed pediatric infection reported according to the CDC. So children generally experience milder illnesses compared to adults, with fever and cough being the most commonly reported symptoms. Early variants, like the original strain and Delta, rely heavily on the host protease TMP RSS2 and ACE protein, which are found mainly in lung cells where TMP RSS2 is abundant. In contrast, Omicron lineage primarily uses a cathepsin-dependent endocytosis route, which is more effective in the upper respiratory tract, where this receptor expression is lower. Thus, we have a variant-specific entry mechanism driving differences in tissue tropism and the clinical presentation. So Omicron will usually present with upper airway predominant inflammation with increased group-like presentation in children. In children, COVID-19 incidence has shown atypical pattern of seasonality compared to other respiratory viruses. And now it follows a winter dominant seasonal pattern like other respiratory viruses such as influenza and RSV, with summer surges having still occurred persistently, influenced by shift in mitigation measures and population immunity. So between fall 2020 and spring 2024, approximately 234,000 children in the US were hospitalized due to COVID-19, with the highest hospitalization rate was seen among children between the age of 0 to 4 years and 12 to 17 years, compared to the school age group between 5 to 11, with pediatric mortality remaining very low, with a percentage of 0.1% of global COVID deaths. So that's the objective of the study, which is to characterize the epidemiology and the clinical burden of SARS-CoV-2 infection in Lebanon, to identify risk factors associated with severe disease and hospitalization, in addition to assess the differences in the clinical manifestation and severity across different COVID variants. So we conducted a multi-center prospective observation study as part of the GIHSN that was explained earlier across 6 different hospitals in Lebanon between the time period between November 2020 and October 2025. And here we selected children under the age of 18 years to be analyzed. So from November 2020 to October 2025, 20,195 patients were screened, with 11,566 hospitalized patients all in all for all age group, and with 6,388 children involved, and with 370 SARS-CoV-2 in 4 confirmed cases. So we ended up with 5.8% of children being infected with COVID. So among hospitalized pediatric patients, SARS-CoV-2 positivity differed by region, with a higher proportion being in the south with 9% and Mount Lebanon with 7.4%, and the lower proportion being in the northern region. And this differences likely reflect regional variation in the hospitalization, mainly pattern, testing practices, referral pathways, rather than a true community incidence. So among the 6,388, as we said, we had 5.8 hospitalized patients with COVID, highlighting the modest contribution of the pediatric population to hospitalization. The positivity rate varied significantly by age group. As we can see, we have children aged between the age of 1 to 5 years were less likely to test positive compared to the preschool age children 5 to 11. There was no significant differences regarding any gender, chronic condition. However, asthma was significantly less frequent among SARS-CoV-2 children, consistent with proposed mechanisms involving reduced ACE2 and TMPRSS expression in asthmatic airways and subsequent inhaled corticosteroids. Approximately 80% of children below the age of 5 years were breastfed, and this was not associated with incidence of COVID. In addition, as expected, the exposure to sick contact was associated with increased rate of hospitalization. To characterize SARS-CoV-2 variant circulation in Lebanon, we retrieved high-quality national genomic sequences from GSAID covering 2020-2025, with dominant clade and lineages were identified for each period, and pediatric cases were classified according to this predominant circulating variant at that time of hospitalization, allowing comparison of age-specific epidemiology and clinical characteristics across variant eras. So after defining the circulating SARS-CoV-2 variant in Lebanon, we examined how this variant translated into pediatric disease temporal pattern. So this figure shows temporal trends in SARS-CoV-2 positivity among hospitalized children across variant period, with the highest positivity rate being detected in the Delta variant and the first Omicron period, with a positivity rate of 25%, with bimodal pattern during the Omicron BA.5. And in later period, we can see the Omicron XBB and JN1 with peak amplitude lower than previously, and with recurrent surges persisting, suggesting a shift toward endemic circulation mode. Notably, pediatric transmission remained active outside the winter season, as seen with consistent summer peaks contrasting with the more defined seasonality of other viruses, for example, influenza or RSV. So using 2021, the Delta period as a reference period, we observed variant-specific changes in pediatric manifestation over time, with malaise being more significantly common in the period of 2024, with wheezes being more detected in 2023, and with gastrointestinal symptoms such as nausea, vomiting, diarrhea were reported more frequently in children. So this table summarizes the demographic characteristics, comorbidities, and hospital course of children hospitalized with the COVID-19 and compared with Delta, with the variant Delta period. We can see that children below the age of 1 year represented a larger proportion of hospitalized patients during the Omicron period. Children with a chronic condition were less likely during the Omicron period to be hospitalized compared to the previous period. And consistent with this, disease severity decreased over time with significantly lower odds of oxygen supplementation and ICU admission during the Omicron period compared to Delta. And this reflects the evolving admission practices and improved outpatient management and reduced disease severity over time. This table also examines the association between a chronic condition and in hospital outcomes among children hospitalized with COVID. And we can see that chronic condition, especially cardiovascular diseases, neurological disorders, and immune deficiency, were associated with higher likelihood of being oxygen supplemented at admission or to be ICU admitted, with prior antibiotic before admission, increasing the likelihood also for these factors. Similarly, the presence of chronic conditions, particularly cardiovascular diseases and neurological disorder, as well as prior antibiotic use and delayed presentation, were associated with higher odds of complication. And notably, patients that received antibiotic prior to admission were more likely, 13 times more likely to die. So among hospitalized children with available vaccination data, nearly all were unvaccinated, with only a small minority having received the vaccine, with a percentage of 2.5%, with a median age of 13.5. Most vaccinated children had underlying comorbidities around 62.5%, and some experienced severe in-hospital outcome, including ICU admission and respiratory support. However, given the very small number of vaccinated patients, this observation should be cautiously interpreted and will not allow to have a conclusion regarding vaccine effectiveness or disease severity. So SARS-CoV-2 accounted for a modest proportion of pediatric hospitalization, with children aged 1 to 5 years were less likely to test positive than those in the preschool age. Asthma was less frequent among SARS-CoV-2 positive children, consistent with prior studies. Pediatric transmission persisted outside the winter season, with Omicron showing bimodal pattern, while Omicron variant overall were associated with high transmissibility and reduced clinical severity. Infants under 1 year of age with cardiovascular, neurological, and immunodeficiency condition remained at high risk for severe disease, with limited vaccination data preventing meaningful assessment of vaccine effectiveness. And thank you. Thank you, Dr.
Thank you, Armani. I think we have a few minutes for any questions for the speakers. All clear? I know we went over time, but thank you very much for attending. Thank you for the speakers. You did an excellent job, and hopefully we'll see you again in 1 year.